Rosalind Archuleta.
Probably not that great. Is it on?
Yes, it's on.
Okay. We're going to go ahead and put a little on my YouTube.
Thank you.
Ms. Archuleta, where do you work?
I work for the New Mexico Department of Public Safety Forensic Laboratory in the DNA section.
And how long have you worked there?
Approximately 14 years.
What education do you have?
I have a Bachelor of Science degree in biology from the New Mexico Institute of Mining and Technology.
And in your current position, did you receive training?
I did. I had approximately 15 months of in-house training at the New Mexico Department of Public Safety. I received my certificate of completion from them at the end of that training program to perform forensic casework.
And do you participate in continual or annual training or education?
I do. Every year I have 8 hours of continuing education credits in the forensic DNA analysis field.
Do you belong to any professional organizations?
I do. I belong to ASS, which is the American Academy of Forensic Sciences, and I also belong to the Southwestern Association for Forensic Sciences.
And in your day-to-day job, tell us just generally what are you doing day-to-day?
On a day-to-day basis, my job is to look at items of evidence for the possible presence of biological material on evidence items collected from a crime scene. When I perform that testing, I run through a series of steps to arrive at a DNA profile. When I arrive at a DNA profile, I then compare that DNA profile from the evidence to any known DNA profiles obtained from individuals associated with the case that I'm working. When I compare the two, I then make a decision as to whether or not anyone is included, excluded, or cannot be eliminated from that DNA profile.
Let's talk a little bit about the Department of Public Safety lab. Is it an accredited lab?
It is.
And how is it accredited?
We are accredited through A2LA, which is the American Association for Laboratory Accreditation, I believe.
And where do they get their guidelines from as far as how to determine whether a lab's accredited?
Right. We follow the ISO 17025 guidelines as well as the FBI DNA Quality Standards guidelines. Those recommendations that are adopted by the FDA Quality Assurance Standards are recommended by SWGDAM, which is the Scientific Working Group for DNA Analysis Methods. In order to use CODIS, we have to follow SWGDAM requirements, and we also have to follow ISO requirements in order to obtain our accreditation.
So there were a lot of acronyms and a lot of things that probably make sense to only a few people. Let's go back for my own benefit. You said the Scientific Working Group for DNA Analysis Methods?
Yes.
Who makes up that group?
I believe it's forensic scientists from, I believe the SWGDAM are, sorry, I'm just trying to remember. I believe they are scientists from the FBI, and I believe they're also A2LA auditors, if I remember correctly.
So these are scientists in your field?
Yes.
And their job is to come up with protocols?
They come up with recommendations for forensic testing.
And then how does the FBI get involved?
The FBI adopts their guidelines and sets forth requirements for forensic laboratories so that we can provide a quality product going forward.
So these guidelines from SWGDAM, the FBI, A2LA, which is your accrediting body,
Yes.
takes those, all those smart people involved in that, takes those recommendations, puts together protocols, and then accredits labs and then ensures that all the protocols are followed?
That is correct.
Did I get that right?
Yes.
Okay. You mentioned CODIS. Can you just tell us what CODIS is?
CODIS is the Combined DNA Index System. The CODIS system has an offender side in which offender profiles are put into the database. As a lab that runs DNA testing, evidence profiles are then put into CODIS from the analyst side of CODIS. And when a sample is put into CODIS, we have to follow guidelines that are set by the FBI regarding requirements to go into CODIS. And that profile is always searched if the profile is entered into CODIS. If we have a hit to a known offender, then we are notified and we let the agency know.
So we're going to get to this in a lot of detail in a minute, but with CODIS, can you put in partial profiles or low-level profiles? What does CODIS require?
We can. They require at least 13 locations. Typically we have more than that, but they do allow some profiles that have lesser information.
And are there requirements on what type of samples you can compare into the CODIS database?
Yes, I cannot put a victim profile into CODIS. I cannot put anything into CODIS that may not belong to the crime scene.
And where does CODIS get these offender samples from?
Different law enforcement agencies across the across the country.
So those are known samples from offenders, and then you're taking casework samples, and if they qualify, as long as they're not a victim and they have the proper number of alleles, which we'll get to, and they fall within whatever other requirements are, you can put them into this database and see if you get
That's correct.
possible match. Now, let's take it back even further. Tell us what DNA is.
DNA is your genetic information. It makes up who you are as an individual. The majority of DNA is the same between individuals. However, there's a very small amount of DNA that gives you your unique qualities. In that small amount of DNA, there is some information in there that doesn't code for anything, and there's a set of small pieces of DNA that I look at as a forensic scientist to arrive at a DNA profile.
Let's talk a little bit about the testing that you do. Can you explain how, when you receive something in your lab, piece of evidence, how do you screen it to decide what and where and how you're going to test it?
When I obtain an item of evidence, I request that item from the evidence unit and have to put my password into our computer software in order to gain custody of that evidence for testing. When I'm ready to work that when I'm ready to work that case and start my testing, I have only one item of evidence at a time. When I open that item of evidence, I'm doing a visual examination of that evidence excuse me, to see if there is anything that catches my attention as far as any visible staining on an item. If there is something, I'll make note of that, perform any testing that is necessary, and move forward. Occasionally, there are samples that maybe I'm unable to see with my naked eye, and so we have something called alternative light source, which lets me look at the item of evidence and see if there is any staining that shows up that maybe I couldn't see in regular light. Once I move forward from that, I collect my samples. I then move to a process called DNA extraction. What that does is it takes that sample that I have retrieved from the item of evidence and it breaks open any cells that happen to be in that tube from what I collected. I'm breaking open those cells so that I can obtain the DNA inside. Once I have that DNA in the tube, I then move forward to a process called quantification. That gives me a ballpark figure of how much DNA is in that sample. From that point, I move on to a process called amplification. This is a process that makes copies of those unique pieces of DNA that I want to look at. Once I do that test, in that step, the DNA is fluorescently labeled. And when I move on to the next step, the genetic analysis of the sample, it runs through a machine called it's a process called capillary electrophoresis. And what that does is it injects the sample. The pieces of DNA are run through a very small capillary within the machine. And as the pieces move through this capillary, they reach a little window inside of the capillary, and there's a laser that shines on that window, and it excites that fluorescent dye. And so as the piece of DNA moves past that window, I see an increase in the signal and then a decrease in the signal as the DNA moves past the window. This gives me the visual representation of those pieces of DNA that I want to look at.
And what else is generated through your testing? You just talked about seeing increases and decreases, but what's the product that you're...
Right, so the product that I arrive at is a DNA profile, or what we call an electropherogram. It is a series of different peaks from different locations on the DNA.
May I approach them? You may. 316. I'm going to hand you what's been marked for identification. It states 316. Do you recognize this?
Yes, I do.
What is that?
It is the DNA profile from Victoria Martens.
Your Honor, the state would move 316 into evidence.
No objection.
316 will be admitted. May I publish? You may. Ms. Marshall, I'm going to put this on our handy, should be on the screen right next to you. So is this one of the profiles you generated in this case?
Yes.
And I'm putting it up here just as an example, but can you walk the jury through, it looks really pretty, it's colorful, but tell us what we're really looking at here.
So what we're looking at are different locations of the pieces of DNA. So if you look at the first location on the upper left, you'll see two peaks labeled 14 and 17. For the individual that was tested, each one of those peaks came from one parent. From each parent. One peak came from the mother and one peak came from the father. If you see in the third row down, you'll see a 7 that is alone. That 7 means that both parents gave a 7, and so I'm seeing that as one peak stacked on top of each other. If I have a DNA mixture in a sample, then I will see more than these 2 peaks at these locations.
So this sample that we're looking at from Victoria is a single source, very clean,
Yes.
easy profile.
Correct.
Just from her, nothing contaminating it. It's a full profile.
That's correct.
What is a presumptive test?
Presumptive test is a serological test that we can perform to help us identify what fluids may be on an item. There is a presumptive test for the possible presence of blood. It is a color change test. If the test is positive, then my result turns bright pink, and I can say that blood was indicated on that item. It doesn't confirm that it is blood. This is why it's called a presumptive test. It does indicate to me that blood is present. And so for me, when I look at an item and I take a sample and I have a presumptively positive test, such as this test for the possible presence of blood that gives me a pink reaction, if I get that pink reaction, then I know that I need to investigate that area further, and I can take a sample for DNA analysis.
Now, once you have an area of interest on an item from a scene, tell us about the types of tests that are available to you in the lab.
As far as DNA testing?
Yes.
So our laboratory uses global filer testing, which tests for the standard autosomal DNA. That's the DNA that comes from each of your parents, the profile that we looked at.
This one, 316?
Yes.
This is a global filer?
That is correct. Yes. And this type of testing can identify down to an individual. I can make comparisons to this type of testing with a known DNA standard, and I can either eliminate someone, include someone, or can make the determination that a person cannot be eliminated or is included in that DNA profile.
Now, are there other types of testing that your lab does?
Yes, we also perform YSTR DNA testing, and that testing looks solely at the male chromosome, and it looks at the male component of a DNA sample. Quite often when we are performing testing, female DNA can overwhelm any male DNA that is present, especially in situations where a sample was collected from someone's person. So in that situation where there's a lot of female DNA, we need a tool to be able to look at that male component of DNA, and that is what the YSTR DNA test does. It's essentially the same type of testing as the global filer testing. However, it uses a DNA platform to only look at that male chromosome information. The difference with this kind of testing is that it cannot identify down to an individual. This is because the male chromosome is passed on from father to son and so forth throughout generations. And it remains relatively unchanged throughout those generations. So any paternal male relatives would have the same male YSTR DNA profile. And so for this reason, if there is a match between a known profile and a Y profile from the evidence, then I can say that that individual and any paternal male relative cannot be eliminated. It can be used to exclude people as well.
What is a buccal swab?
A buccal swab is a known sample from an individual. It is a DNA swab that is collected from the inside of the cheek or the buccal cavity.
Does it look like a Q-tip?
It does.
What are extracts?
DNA extracts, they are the end product of the DNA testing. When we conclude testing in whichever form it is, whether it be global filer or Y filer testing, the extract is then documented as to the volume that we have left, and then we dry that sample down in the event that maybe we need to move forward with further testing on that sample.
So you can take an extract and use it later for more testing?
Yes.
Let me back up a little bit. You talked about global filer and Y filer.
Yes.
Are there other DNA kits out there?
There are a good number of different kits, but our laboratory is accredited for using the 2 kits.
How come you can't just get all of them?
We need to perform validation testing on everything to make certain that the tests that we employ at the laboratory are reliable, give us reproducible results, and accurate results. And so that is the reason that we need to perform that validation testing. And so we can't just use any tests that we can buy off the shelf or from a different company, just because there's a whole series of steps to put into place in order to even use that kit for casework testing.
Now, if you were to leave the Department of Public Safety and go to another lab and have different kits, could you just start testing on those kits?
No, I would need to learn about the kit. I would need to go through different training and learn how to report and interpret those results.
And is part of your ongoing training maintaining your proficiency in the kits that are online at your lab?
Yes, it is. We undergo proficiency training 2 times per year in our DNA lab.
Going back to DNA generally, can DNA be destroyed or degraded?
It can. DNA doesn't like extremes. So if DNA is exposed to high heat, moisture, bacterial or fungal contamination, cleaning products such as bleach, that will break down DNA. UV light. There are a lot of different factors that can break down DNA.
Can DNA be cleaned off of something?
It can be cleaned off of things. It can be, I suppose, wiped off or cleaned off using products. If anything, I would expect a lesser amount of DNA on an item, but if somebody is very thorough, it's possible to not find any DNA as well.
Can you or anyone tell when DNA got onto an item?
No.
Can small amounts of DNA be obscured by bigger amounts of DNA?
Yes, as I spoke about before, occasionally when a sample is collected from an individual, I would expect to see that individual's DNA present. So, for example, if I took a swab from my hand because I was braiding my daughter's hair or something, I would expect to find my DNA on that sample. But if I have touched someone, like my daughter, I wouldn't expect to find a whole lot of her DNA there. It's possible, not a lot, sometimes none. But if there is other DNA there, it wouldn't surprise me to find that my DNA overwhelmed that small amount of DNA.
And by overwhelmed, what does that look like in the result that you would get?
Oftentimes it would look like my DNA profile alone. If there's enough to be detected, sometimes it may look like a very small amount of DNA that maybe is a minor component or contributor to a DNA mixture.
I think we talked about this a little bit, but I'm going to put this back up, 316. What are alleles? What does that word mean?
So alleles are those pieces of genetic information that come from each parent. So each allele, you'll see on the upper left under, I think it's a green header, it's kind of hard to see right here, but yes. That 14, that 17, each one of those pieces are what we call an allele. That information is a piece of DNA that has a repeating sequence. So the number that is given to that allele is the number of repeats of that DNA that we see.
Do DNA tests always come back with something you can compare?
No, not necessarily. Occasionally we maybe may find that there is no DNA profile, even though we were able to detect a very tiny amount at the quantification step. And occasionally we may have a profile that just doesn't have a lot of DNA, and maybe we need to call that profile uninterpretable because there is just information that is just not strong enough to get a full DNA profile, such as the one you see.
What about mixtures? Tell us about mixtures. You talked a little bit about your example with your dog and your hand, but tell us about mixtures.
So a DNA mixture is a profile of DNA from more than one individual. So if you use the example with my daughter, if I take a drink from a glass of water and then my daughter comes and takes a drink of my glass of water, I would expect to see my profile there, but now I'm also going to see hers as well. And so, I am going to see more than just those 2 alleles at those locations. I would also expect to see DNA from her as well. And so at each location in a DNA mixture, I may see 2, 3, 4, maybe more alleles at that location, indicating to me that there's more than one individual present in that sample.
Are there times that you can pull out just your profile and separate it from your daughter's profile?
There are times in which someone is a stronger, oh thank you so much, I'll get one in a minute, in which someone might be a stronger contributor to that DNA mixture. In that case, I can see who the major contributor is because they're present in higher amounts. And then there's a minor or the lower contributor to that mixture that you can see in there, but maybe there's not enough information to interpret that minor profile. So if there is a major contributor, I can then pull out that major contributor and I can then make a comparison to that major profile.
Are there times when there's mixtures that you can't separate like that?
Yes.
And what happens with those?
It depends on the type of testing that is being done. With global filer testing, I have to look at that profile and make a determination about a lot of different things, whether or not there is enough information present, and if there is enough information present, I can then make a comparison to any known standards. If there's not, I may issue a report stating that maybe it's insufficient or partial, for example, which means that some of that DNA information maybe isn't present. But if there is a association, I will then make that comparison, decide who might be eliminated, who might be included, and run any statistical calculations that may be necessary.
When you're saying insufficient, is there guidelines as to when something's insufficient?
Yes.
And why are those guidelines in place?
There are guidelines in place within our protocols that allow us to look at a DNA profile and make a determination as to whether or not very low-level information may be noise, may be good information that is not strong enough for me to see a full signal from. And so if that is the case and it falls within our thresholds, then I'm not going to use that information for statistical calculations. If I don't use that information, then my statistical calculation will be a little bit more conservative.
So going back to this, what we've determined, nice strong profile from Victoria, when you're talking about these low levels, we're still going to see some boxes and spikes and stuff on the graph, right?
That's correct.
But you're saying that they're not reliable. Is that fair?
Yes, if there is below threshold information, it will be marked, and the profile will have a note, or the statement will read that the sample is insufficient.
Do you use those to make any comparisons?
No.
Why not?
I don't want to falsely include or falsely exclude someone by making a comparison to information that may not be reliable.
Going back to mixtures just briefly, when you get a mixture, how many people can be in a mixture? We talked about 2, but...
Any number of individuals could be in a mixture.
And as we add people's DNA in a mixture, does the analysis that you go through get more and more complicated?
It gets more and more complex with each individual that is included in a DNA mixture.
Your Honor, at this time we would like to qualify Ms. Archuleta as an expert in the area of forensic science, in the area of forensic science of serology and DNA analysis.
No, Your Honor.
Should be permitted to testify in that capacity? Ms. Archuleta, did you analyze DNA in the Victoria Martens case?
Yes, I did.
How many tests did you do in this case?
The number of samples total that I ran was 323.
Of that 323, how many were the standards, the buccal swabs, or the known people?
I believe it was 38 samples were known samples, which leaves approximately, I believe it's 285 evidence samples that were examined.
Were some items of evidence tested more than one time in this case?
Yes.
Would an example of that be hairs? Or actually, not hairs, I'm sorry. Would an example of that be body standards or other items of evidence like things from the scene?
Yes.
Were there things that you tested, going back to my hairs, you tested dozens of hairs, right?
Yes.
Is this the largest case you've ever done?
It is.
Is this the largest case that the state lab, as far as you know, has ever done?
As far as I'm aware, yes.
And did you get, we talked about kind of the variety of results that can happen. Did you get a variety of results in this case?
I did.
Did you get matches where you were able to match a known standard to something from an item of evidence?
Yes.
Did you get cases where people cannot be eliminated
Yes.
from an item of evidence?
Yes.
Did you find mixtures?
Yes, I did.
Did you find insufficient or uninterpretable pieces of DNA?
I did.
And you talked a little bit about statistics. Is that only something that's done when you have those good comparable samples to a known standard?
Yes. If there is an association between a standard and an evidence profile, I will then run statistical calculations on the evidence profile. If it is a single source profile, I will run random match probability calculations and report the most conservative number out. If it is an inclusion, but is not a major profile, I will run what is called combined probability index calculations, and those results will be published with a graph or a range of frequencies for that sample.
I sort of understand what you're saying. So let's break that down.
Sure.
So for the combined probability
Inclusion.
what in lay people terms, what does that tell us?
So the combined probability of inclusion statistic is a statistic that lends weight to the association of the match to that profile. So if I have to run these statistics on that combined probability, then I can, with confidence, say that someone is included as a contributor to that profile. But the range of numbers that is published in the report gives me an indication of how strong that association is. So if it's a very high number, then that's a very rare profile. I wouldn't expect too many other people to fit into that DNA profile. And I would have to test a lot of random, unrelated people from the general population of New Mexico to find one other person who might also fit into that DNA profile. But if you see a number that is maybe 1 in 10, I could test 10 people in this courtroom, and they may also fit into that DNA profile. So that would be a very common profile if the number is very low.
What about random match? Tell us about that.
Random match probability gives us an indicator of how strong, again, that association is with that match to the single source profile. Again, if it's a very large number, in my opinion, I wouldn't expect to see another person with that DNA profile unless they had an identical twin. Identical twins will have the same DNA.
So I think we've all watched, the first 48 and shows like that where there's some analysis going on, and I've heard numbers bigger than I can say in some of these statistics. So when you get those huge numbers, is that saying that it's unlikely probably anyone in the world? If it's bigger than the population of the world, very unlikely we're going to find anyone, right? So that would be a very strong example.
Yes.
All the way down to sometimes you get very low ones, 1 in 10.
Yes.
So that's just telling us the odds of anybody else having these.
Correct.
And does Y testing utilize a different method for computing statistics?
Yes. If I have an association within Y filer testing, I run the statistics for Ys, and that is done via the counting method, which means there is a database of published Y profiles, and the counting method looks at the number of times the evidence profile that I enter is encountered within that database.
Let's move to the evidence in this case. How did you receive the physical pieces of evidence in this case?
All of the items are delivered to our laboratory by the law enforcement agency. Once the agency is dropping off the items of evidence, our evidence section will give the case a unique case number, log in those items of evidence, and put them in a secure evidence vault.
In this case, was that typically Detective Brown or Detective Lewis with the Albuquerque Police Department?
It was, yes.
Okay, and once you receive those?
Yes. When I'm ready to start my testing, I then request those items of evidence from our evidence unit. In order to gain custody of those items, I have to enter my password into our laboratory computer system that maintains our chain of custody for the items. And then I move those items to my locker, which only I have the key to, and the only other person who has the key is my supervisor, just in the event of an emergency. When I start my examination on those items of evidence, I can pull those items out one by one for visual examinations.
When you received the evidence in this case, was it properly labeled and sealed?
Yes, I do recall one item starting to come open at the top, but I did remediate that seal and put my initials and date over it so that nothing could fall out or enter into the bag.
And were there signatures on the seals that were made by you and other people?
Yes.
And sometimes multiple seals and signatures?
Yes.
In your lab, do you get the first go, or is there another unit that if specific testing is required, they go first?
If other testing is required, the section that will perform testing first is latent prints. That is because latent prints are fragile, and they use methods that are safe for DNA, so that when they conclude their testing and they pass the items of evidence to me for testing, I know that I can do my testing and that DNA on the item of evidence hasn't been harmed by their testing.
I think you talked a little bit about this already, but the protocols that you have in place to ensure that items are not contaminated, you said earlier that you open one thing at a time, you're wearing protective gear. Do you follow all of those practices in examining evidence?
I did, yes.
And after you opened something up and tested it, was it resealed and signed by you and put back into evidence?
Yes.
And I think we talked about extracts, but are those items of evidence essentially that you created in this case?
Yes, they are.
And those get tagged as well, labeled, and put into evidence?
Correct.
To jump ahead to the very end of this, those extracts were those at one point provided, or at least some of them provided to an outside DNA lab called DNA Labs International in Florida?
When I was done testing, the agency picked up the items, and the agency then submits samples to whatever other location he's going to submit them
So
to.
you're aware that some of your extracts.
Yes.
When you test items of evidence or DNA standards or samples, do you write reports?
Yes, I do.
Who reviews your report?
Our reports all go through technical review as well as an administrative review. The individuals who are qualified to do that are other qualified forensic scientists in the DNA unit. An administrative review can be any forensic scientist within our lab.
And why do you need your reports to be reviewed?
We want to make sure that when the analyst that is writing the report, that everything is done properly. We want to make sure that our protocols have been followed, which is what the technical review looks for, that the appropriate testing has been done, and that there aren't errors throughout the throughout the reports or the case notes. That way, we know the results that are going out are accurate.
You talked about case notes. In a typical homicide in 2016, about how many pages would you generate?
Anywhere from 1 to maybe 300 pages.
How many pages were generated in this case?
Over 1,800 pages.
And how many reports did you write?
13.
And did some of those reports have amendments?
Yes.
You said 13. Was it 13 or 12?
I believe it's 13.
And why were there some amendments?
There were small typographical errors in them.
None of your conclusions changed?
No, none of my conclusions changed.
When you got this case, did you have Victoria's DNA standard?
Yes.
And what about Michelle Martens?
Yes.
Fabian Gonzales?
Yes.
Jessica Kelley?
Yes.
And then you talked about 38 other standards or total standards. Did you receive standards from people like Victoria's little brother and males associated with this case?
Yes, I did.
Before we dive in, when you get 38 different standards, and you've already, those came in not all at once, correct?
That's correct.
So when you get a new standard, you have to go back and compare it to everything you've already compared?
Yes, so when I get additional DNA standards in a case, I run the DNA testing on that item, so I have a DNA profile for that individual, and then that DNA profile is compared to every item of evidence that has previously been tested.
So as we discuss relevant items, can the jury feel very comfortable that you've cross-checked, so to speak, every possible item to everybody who's
Yes.
used? When you find unknown yet comparable DNA on an item of evidence, did you compare it to other unknown yet comparable DNA from other items of evidence?
Yes, I did.
So an example would be if you found two unknown yet comparable Y profiles, you compare them to each other and see if that was the same person.
I did.
I think it's fair to say, and tell me if you disagree, that it would take us probably a few days to go over all 200 and whatever that number was
Yes, it would.
tests that you did. So we're going to move to just what is relevant in this case. So did you receive knives in this case?
I did.
How many knives did you examine?
I believe the number was 12.
Multiple knives?
Multiple knives plus a knife block.
So you received a knife block with knives in it and some...
And some that were packaged separately.
And did all these knives go through your presumptive screening looking for biological material?
Yes,
And did you get results on one of those knives?
on one of them.
If you need to, Ms. Archuleta, grab your case file
Thank you.
and your clips to that. You're hooked up. So going to the knife that you generated a result on, which I believe is from your July 20th of 2017 report, it's 1834A1.
Yeah, may I refer to my reports? Thank you. I'm sorry, can you repeat the date?
Sure, July 20th of 17. And it's 1834A1. I'm going to put up what's been admitted as state 61.
I'm sorry, you said July 19th?
July 20th.
July
17.
I'm sorry, are you referring to April 20th?
Probably.
You're
Yes,
referring to the
April
report with the
20th.
knives?
Sorry, mine is a typo. It was April 20th.
That's okay.
I apologize.
That's all right. Yes,
Sorry.
that's all right.
Trying to be organized. There's a lot. So, April 20th report, 1834A1. And 1834A1, is that a number, so when things come into you, they have APD numbers?
Yes, at the time, the agency was giving us their item numbers that they created for the item that was submitted. If I had to take multiple samples from that item, I would then sub-itemize that. That may be in the form of a -1, -2, maybe an ABC. It just depends on the item and how the agency had labeled that item prior to it coming to the laboratory.
So this item that you received was labeled K1 with some other knives, is that right?
I had this item as 1834A1.
Is that because it went to Layton's first and that's the GPS number?
1834 is the item number that the agency gave us. So when I opened the item, there were multiple items inside, so I labeled them with the item number and then A through M.
Understood. So do you recognize, I put what's been admitted as state 61. Do you recognize this?
Yes, I do.
Is this the knife that we're talking about?
Yes.
1834A1?
Yes.
And can you tell us what results, well first, what did you notice about this item of evidence?
I noticed the red-brown staining that's at the juncture between the blade and the handle.
And is that an area that you focused on?
Yes.
What results did you generate from this item?
On 1834A1, which is the swab of the knife blade and the possible biological tissue from the blade handle and juncture. Blood was indicated on that item. And the DNA profile obtained from 1834A1 matched the DNA profile from Victoria Martens. All other standards that were submitted were eliminated.
You also examined some clothing items recovered from the scene. So, directing you to 1836A, which would have been from your, I believe, March 8th report?
Yes.
Of 2017? Yes, I'm sorry, March 8th of 2017. I'm going to show you states what's been admitted as 36, and this is a scene photo of this item marked CL3, and then also states 40. Does that item look familiar?
That item does look familiar.
So, I believe this item is actually 1836C. Can you
I
tell us about your screening of this item? How early on in your testing did you receive this item?
So I did receive this item early in my testing, and on the March 8th report, I'm sorry, you're talking about item 1836?
1836, so I think you received some items packaged together.
Yes,
Some clothes,
it was clothing from a trash can in the kitchen in the apartment, and I labeled item 1836A as underwear.
Okay, and I believe this is C. Is that right?
Yes, and I think that was done on a different report.
Okay.
Yes, it was done on the May 24th report, 2017.
So go back to just when you received this jumper. Let's
Yes.
go to this jumper. These were done fairly early in the testing.
That's correct.
Up here. And this is confusing because you have 15 or 16 reports, right? So
Yes.
we're going to try and get through it, but I'm going to try and direct you, but hopefully my notes are right. So generally in a case, when you get an item of evidence like this, would you swab something like this just blindly? What are you doing here?
No, I'm looking for any biological material on the item. Given the case information, excuse me, given the case information, I was looking for any blood that might be present and possibly any semen that might be present on the item.
So early on in this case, back in early 2017, when you're screening these items, would there have been a reason for you, would there have been some evidentiary, well, let me back up. Would you have needed some evidentiary basis to swab a certain area aside from your screening?
Yes, typically an agency will let us know if they're looking for maybe touch DNA on an item, and that would indicate to me that maybe I would need to take a general swab of the item. That wasn't done for this item, and so at the time I tested only for the possible presence of blood or semen.
So there was no information at that time provided to you to hone in on a specific area of any of these clothing items found in the kitchen trash?
No.
So just going back to 36, You said that 1836A, which should be from your March 8th, 2017 report, was a pair of underwear,
Yes.
and they're depicted here in 36. I know that's not your photo. Do those look familiar?
It's hard to tell from that photo.
Fair enough. This item was described in this trial as being soiled, which sounds like it had urine or feces or something on it. Tell us about your screening and what you found on the underwear.
On the underwear, I tested for the possible presence of both semen and blood, and no semen was detected and no blood was detected.
What result did you get from the underwear?
Human male DNA was identified on 1836A, which was the underwear. However, it was not in sufficient quantity for further DNA testing.
Is that an example of sort of just that lower level amount?
It is.
What about an orange and white sock, which is 1836B, also located in the kitchen trash?
Yes, 1836B. There was nothing of serological significance detected on that item, so I did not move forward with any other testing.
And then we talked a little bit about this jumper. Tell us what result you had from the jumper 1836C.
On item 1836C,
Which I believe is March 28th report.
yes. There's a lot to read through, sorry. No semen was detected on item 1836C.
Was there a subject for blood on this?
Yes, and that was also negative. No blood was detected.
And then there was a white sock, also 1848A.
And this was on which report?
This is from the June 2nd, 2017 report, 1848A.
Yes, clothing from trash can and kitchen.
Correct.
Okay.
The white sock. What result did you get? I believe you had 1848.
Yes, I had 1848, and I sub-itemized that into two items. On 1848A, the swab of the outer surface of sock.
Why did you swab the outer surface of the sock?
To my recollection, there was staining on the outside of that sock.
And what was your second sample?
The second sample was inside of the sock. It was the inner sole,
Why did you do that?
to see who might have been wearing that sock.
And what result did you get?
On 1848A, no blood was detected. And on item 48B, no blood was detected.
Did you move forward on either of those samples?
Yes, I did.
And which one?
On item 1848A and 1848B, no human DNA was detected.
Now, no human DNA was detected. What does that mean?
That means either I did not find any human DNA on it because it wasn't there, or it was there in very, very low quantities.
Moving to some items found on top of the dryer, which I believe are going to be in your April 20th of 2017 report, and then lies in your June 2nd, 2017 report. There's a Tinkerbell comforter labeled 1832B.
Yes.
And it published states 28. These items. Going to the Tinkerbell comforter, tell us about your testing, what you found.
On 1832B, the Tinkerbell comforter, I did take a cutting of a stain, which I sub-itemized as item 1832B-1. Blood was indicated on item 1832B, which was the comforter. No semen was detected on item 1832B. Human male DNA was identified on 1832B-1.
Is the -1 the cutting that you made?
It is.
And let me just ask you, you said human male DNA was identified. Is this during your autosomal or the global filer testing?
This is during the quantification step to find out how much DNA is in that sample.
So in the quantification step, you're seeing human male.
Yes.
So you're only seeing just half of the whole DNA profile, is that right?
Well, I don't know that at that point.
Okay. So what did you do once you saw human male DNA?
I did move forward with testing on that item. I'm sorry, I'm just reading through here. I moved forward with the standard autosomal testing through Global Filer, and the DNA profile that was obtained from item 1832B-1 matched the DNA profile from Victoria Martens. All of the other standards that I had at this point were eliminated.
Did you do Y testing on June 2nd?
Yeah, I did move forward with Y testing for 1832B-1, and I found no male YSTR DNA profile was obtained from that item.
So you saw male in the quantification step, but then did not produce the results?
Yes.
And the testing that you did produce the results on was the 1832B-1, which also had a presumptive blood screen?
Let me go back to that just to remember. That's okay.
That's that first. From 420.
1832B,
-1.
yes, blood was indicated on that.
So that was the sample you took. You got the presumptive test for blood that we talked about. You went forward and it matched Victoria.
Correct.
So would you say Victoria is the source of that stain?
In my opinion, yes.
There's also a Rugrats fitted sheet, which is labeled 1832D-1, which I believe you tested on April 20th.
Yes.
And we're seeing that also in photograph 28, found
Correct.
top of the dryer. Tell us about your results there.
On 1832D-1, I did take a cutting of the stain from that sheet. On 1832D-1, blood was indicated on that item. No semen was detected on that item. And then I did find that human male DNA was identified on 1832D-1. At the time of testing on the April 20th report, an unknown male DNA profile was obtained from item 1832D-1, with Jessica Kelley, Fabian Gonzales, and Michelle Martens and Victoria Martens being eliminated.
And then later on, we talked about going backwards when we get more samples later. Did any of the other standards you received match up to this?
Yes. The DNA standard for Matthew Oding matched that DNA profile.
And that's Victoria's little brother?
To my knowledge.
And Matthew matched the Y that you tested, is that right?
Yes, I believe so. I was looking at the Global Filer results. I can verify that.
Should be on June 2nd.
Thank you. That is correct.
So just to discuss a little more, 1832D-1, this fitted sheet, there was a place that you thought possibly contained biological material in your screening process. You presumptively tested it. It tested positive for potential blood. You went forward and you eliminated Jessica, Fabian, Michelle, and Victoria.
Yes.
Found a Y, which is male, went forward, and it ended up being a match to Victoria's little brother.
Yes, it was an unknown male DNA profile at the time.
For a little while, until you got the
Until I had the standard.
So actually, let me put this picture back up. I don't know if you can see it very clearly, but this dryer door is ajar, and there's probably a better picture already entered into evidence, but those two items came with a bunch of other items. Do you recall that?
I do recall there being a bag from the dryer that had many, many items in it.
And all of those items you screened
Yes.
with nothing, and you didn't find anything with serological significance on any of those other items. Is that right?
I believe that is correct to my recollection.
Let's move to your, and I'm sorry to jump around, your reports on April 20th. There's a green hand towel from the coffee table in the living room, which is labeled 1847A.
Yes.
So I'm going to show you states 106. It's a little washed out, but looks a little white on the screen, but that's a green towel. So tell us about the this item, 1847A, what did you tell us about your results and your testing?
On item 1847A, blood was indicated. No semen was detected on that item. Just reading.
That's okay.
There are a lot of numbers I don't want to transpose, so I'm trying to read carefully. Human male was identified on item 1847A. I did move forward with DNA testing. The DNA profile obtained from item 1847A matches the DNA profile from Michelle Martens.
Anybody else need a pen or paper? Pen? 2 pens. Okay, so back to our green hand towel, 1847A. You said the DNA matched Michelle Martens?
That is correct.
Did you move forward with any other testing?
I did perform male YSDR DNA testing.
Tell us about that.
A partial male YSDR DNA profile was obtained from 1847A. However, no interpretation or comparisons can be made due to an insufficient quantity of DNA in the sample.
There were some items collected from inside the washing machine, which I believe are reflected in your April 20th and also June 2nd reports. I'm going to show you what states 24. Those are, there's 2 towels, a white towel, and then I'll call this a beach towel. A green, it says blue, green, and white bath towel, just we'll call it that. So the white hand towel is 1833A. Tell us about the testing you did on that towel.
On the white hand towel, 1833A, I took 3 separate cuttings from that item.
And let me just ask you, when you say cuttings, those are spots that you think might have some biological material on them.
Yes, when I perform a visual examination on an item of evidence, I can collect it a couple of different ways. I can either take a swabbing of that item using a sterile swab and sterile water. I would moisten the swab and then swab with the staining on the item. I could take a scraping of the item or maybe scrape off any surface material to collect some of it and put that into a tube. Or I can take a sample of the item itself, just a small cutting of whatever the item is. For example, here it's a towel, and so I took a small cutting of the towel.
And we had retired detective Pisano testifying. He actually held up this towel and I saw little squares cut out. Would that have been your work?
Yes.
So let's go discuss 1833A. A1.
Okay. On item 1833A1, blood was indicated on that item. Human DNA was identified on item 1833A-1, however not in sufficient quantity for further DNA testing.
Did you look for male DNA? Well, let me ask you this way. I have from your results, it says no human male DNA. What does that mean?
That means I was not able to find any human male DNA that was detected at the quantification step.
So you were looking for male, didn't see it. Does that mean any DNA there is female?
It's possible, or it means that I was not able to detect all of the DNA because it was in such low level. I can't say anything about it.
So we just don't know?
I don't know. It's,
So
to my knowledge, based on that test, it is human DNA that was identified, but I can't say anything about what it might have been.
So your
as far as
report
male
says
female.
no human male DNA, but that's not excluding that it possibly could be male. You're just not seeing enough.
Correct.
So let's go to 1833B, which I believe was also on your June 2nd, starting on your April 20th, and then your June 2nd report. 1833B.
Yes, the blue, green, and white bath towel.
Yes, I have 1833B1 is listed as a stain. Can you talk about that?
Yes, on 1833B1, on 1833B, the entire towel itself, blood was indicated on that towel. On item 1833B1, human male DNA was identified on that item. did move forward with testing. I found a partial DNA mixture of at least 2 individuals that was obtained from item 1833B-1. However, no interpretations or comparisons can be made due to an insufficient quantity of DNA in the sample.
You said it's a partial mixture, so describe what that
Right.
means.
So you'll recall that I said when you have a mixture of DNA, you see multiple pieces of information at each location for the DNA that we're looking for. Occasionally, when there is a very small amount of DNA present, I might see that information missing from some of those locations. And so I have reported it as a partial DNA mixture.
So you didn't have...
I didn't have all of the information across the profile.
And did you perform Y testing on 1833B-1?
Yes, I did.
What was the result there?
Is a partial male YSTR DNA profile from item 1833B-1. No interpretation or comparisons can be made due to an insufficient quantity of DNA in the sample.
So just discussing this result and the one we talked about a minute ago where it said no given male DNA. Here you're finding a partial male. Can we say, or can you say, if this is male or female, or you can't tell?
I'm not able to make a comparison about that. Regarding the male YSTR
Right.
you're referring to, sorry. I would say that that is male DNA, and the reason that I say that, at least some of that DNA in that sample is male DNA. The reason that I say that is because when I test for Ys and I use the Y-Father kit, that test is designed to detect the male chromosome information. And so if I am seeing information, there is some male information there.
So even if it's low level and you can't compare it to anything, you know that it's at least male.
Yes.
Versus our last result where it says, I think it was in the quantification stage where you said no male detected.
Yes.
That is a different result than this. Is that
Yes.
fair? You also listed 1833B2, and that's listed as possible biological material.
Yes.
Tell us what that was.
As far as the result goes?
If you remember just when you say we talked about stains and now you're talking about just biological material, what did you find on this?
Yes. What I found, to my recollection, was a small tangle of hairs that looked like it had some sort of, what looked to me like biological material in it. It was a small piece of a yellowish material that was delicate and a slight bit greasy or oily, which made me question as to whether or not it may be of biological origin. And so I did take that piece of possible tissue and I moved forward with further testing.
Tell us about your results on that.
And that was 1833B-2. Again, on item 1833B, the entire item, blood was indicated on that item. Human male DNA was identified on item 1833B-2. The DNA profile obtained from item 1833B-2 matches the DNA profile from Victoria Martens.
So we have a little piece of potential human tissue or body tissue, and that matched Victoria, but you're also seeing human male. Can you
Yes.
explain how we get both of those?
Again, it's possible that that male DNA is just present in a very low amount, so I'm not seeing that.
And did you take a little piece of the towel with that biological material?
I believe with the biological material, I only took a sample of the material itself, the biological tissue.
So it's a match to Victoria, and then there's a low level of male DNA present in that same sample?
Yes.
I'm going to move to the carpet from Victoria's room, which I believe is reflected in your, actually, it's in your April 20th and then also June 2nd Y report. But just generally talking about the carpet. Did you receive a whole rolled up piece of carpet?
I did.
I'm going to show you 206. Does that appear to be consistent with what you remember?
Yes.
had some carpet fibers and then a cutting.
Yes.
Were both of those presumptive of blood?
Yes.
Were you able to match Victoria to both of those samples? Was she the contributor to both of those samples?
Yes, she was.
And you ran statistics on there on
Yes.
both of those and confirmed that Victoria was indeed the source of both the cutting of the stain area and the fibers.
It matched Victoria Martens, yes.
There was some other potential blood collected from the area around Victoria's room. And these samples, I believe, came to you in the form of blood swabs taken by police. So let's move to we're still on your April 20th, 2017 report, B16.
2678 B16.
It is 2678B-16.
Okay.
I'm going to show you Stage 130, which is a scene photo of B16, the light switch. So what you received is not this light switch. You had the little Q-tip.
That's correct.
That we heard Detective Pisano describe. The swab they took, put in an envelope, snapped off the end, and then you get the little envelope, right?
That's correct.
Okay, so let's talk about 2678B-16, this light switch cover. Tell us about your results on that.
Blood was indicated on 2678 B-16. No human male DNA was detected on item 2678 B-16. And no human DNA was detected on item 2678 B-16.
I'm going to put up 131. Oops, too close. This is just a closer shot of B16. So your presumptive test was blood indicated.
Yes.
And we're seeing what appears to potentially be blood here on this light switch area. How can you explain not getting any human DNA or any, what does that mean?
Firstly, I don't know where the sample on that light plate was collected from. Secondly, what I see from that image is some dilute staining. Depending on where it was swabbed and how much of it was collected onto the swab, our serological test for the possible presence of blood is very sensitive. And so I may see a color change indicating that blood might be present there, but there still may be a very small amount of DNA present on that sample, and I might not be able to detect that moving forward with testing.
Generally speaking, if an item of evidence is cleaned or wiped, is it possible to get a result like this?
It is possible, yes.
Let's move to B-18, which is, I believe it's also 2678B-18, and it should be on your July 19th of 2017 report.
Yes.
Tell us, and this is indicated as the right side edge surface of entryway wall from living room to hallway. Can you tell us, and I'm going to put up 173, showing the marker from the scene of B18.
Judge, may we approach?
And I think that we left off on B18. Is that your recollection?
Yes.
Okay. So B18, can you give us a description that's in your report?
The description that is in my report are swabs of staining, which was represented as from the east side of doorway between bathroom and living room.
And your numbers, 2678, and then you have B-18, does that correspond with the scene marker? Or the numbers you received on the labels from APD?
Yes.
So tell us about 2678B-18.
Blood was indicated on 2678 B-18. I moved forward with testing that item. Human male DNA was identified on item 2678 B-18. I found a DNA mixture of at least 2 individuals from item 2678 B-18. The major DNA profile resolved from the mixture matches the DNA profile from Michelle Martens. Could not make an interpretation or comparison regarding the minor contributor to the mixtures due to an insufficient quantity of DNA in the samples. Jessica Kelley, Jessica Kelley, Fabian Gonzales, Victoria Martens, and Matthew Oding are eliminated as sources of the major DNA profile resolved from this mixture. With the other DNA standards, they were also eliminated.
So you have a mixture with a major profile that's Michelle Martens, and then a minor that was not comparable?
That's correct.
Let's move to B19 or 2678B-19, which is in our April 20th of 2017 report.
Okay.
Can you read the description of that item for us?
Swabs of staining from north door jamb of northwest bedroom.
Can you tell us what your results were for 2678B-19?
Blood was indicated on 2678B-19. No human DNA was detected on item 2678B-19.
Let's talk about the wipes container found in Victoria's bedroom, which I think is reflected on your May 22nd, 2017 report, as well as the June 2nd, 2017 report. Do you remember receiving this physical item of evidence?
I do. Sorry, I'm just going to look for it quickly.
If I showed you a photo that you think you'd recognize it?
Yes.
I'm going to show you states 168. Is this the item that you received?
Yes. I'm sorry, you said May 22nd, 2017?
That's what I wrote down. I believe that's the right one.
Yes, that one was replaced by the August 8th report.
Okay, that was one of the ones with the typo?
Yes.
So it got replaced by August 8th?
Correct.
So if I say May 22nd again, we need to go to August 8th?
Yes.
2017? Okay. So let's talk about this wipes container. Labeled 2674, and you have some sub-items A and B. Tell us about the testing that you did on this wipes container.
On the wipes container, I took two sub-items. 2674A was a red-brown stain from the inside lid of the wipes container. 2674B was a swab of the outer lid of the wipes container.
So you said the inner lid
Yes.
and the outer lid. So looking from the zoom in, it's a little hard to see, but this container looks open. So you're talking about this would be the inside, and then the other side, if it were closed,
That's correct.
what you label outside.
Yes.
Go ahead and tell us what results you obtained on this item.
On 2674A, which was the swab of the staining from the inside of the lid, blood was indicated on that item. Human male DNA was identified on item 2674A. The DNA profile obtained from item 2674A matches the DNA profile from Victoria Martens. And I'm sorry, did you say the Ys were on the June report?
Yes, June 2nd. Before you move to June 2nd, on your August 8th report, was male DNA identified?
On 2674A?
Correct.
Human male DNA was identified on 2674A.
And was that in a sufficient or insufficient quantity to move forward?
It was not in sufficient quantity for further DNA testing.
So we have blood so you took your own swab here.
Yes.
You have a blood indication, the profile matches Victoria, but we also have this sort of male DNA that's just at an insufficient level.
That is correct.
Okay, let's talk about, let's see, I think that's it for that one. And you eliminated, it's basically a match. Victoria's the source, is that fair?
Yes, Victoria Martens matched that DNA profile.
Okay, let's go to 2674B, also on your August 8th. So this is now the outer lid.
Yes.
If it were closed, it would be this.
A DNA mixture of at least 3 individuals was obtained from item 2674B. However, no interpretation or comparisons can be made due to the complexity of the mixture.
That's one of those ones we talked about, just too much there.
There was too much information to make sense of it.
Okay, first witness from TV. I'm going to show you what's been marked for identification in states 317 through 321, and just tell me if you recognize these items.
I do.
Okay, and what are these?
These are images of item 2675, my markings for ID on the outer package, as well as the item inside with my markings on the item itself, and of the areas that were tested.
And this is a blanket
Yes.
that you tested in this case?
Yes.
Your Honor, the state would move for admission of states 317 and 321.
No objection.
317 through 321 will be admitted. May I publish? You may. Ms. Archuleta, let's move to item 2675 that we just saw some pictures of. And that should be from, I believe, your 8/8/17 and also 6/2/17 report. So I'm going to publish what's been admitted as states 317. You just identified this. Out. I'm actually going to put this way. So tell us what we're seeing here.
That is the outer packaging of item 2675, and the blue markings to the left of that label are my markings for ID with the case number, the item number, my initials, and the date it was examined.
And inside that packaging, we have this item. Do you recognize this?
I do.
I see some blue writing. It may be a little hard to see, and I'm going to show 319 to also sort of show what I'm talking about. Okay. So I see some writing on here. Is this something that you do during your testing?
It is.
Can you do that on all the items that you test?
Most often, yes. Occasionally there are items like this item I was having a difficult time marking it just because of the fleecy surface. Some items just don't lend themselves well to a marker.
But in general, as you're
In general, yes, I do.
You're trying to mark where you've taken a sample, and then you're trying to put your initials on there. So
Yes.
whoever opens that item of evidence later will know that you've been there.
Correct.
I'm going to show you 320, actually 320. And I see a blue circle. Is that your blue circle?
Yes.
And a little thing? there.
Yes.
And let me show you 321, which might be a better photo. Another blue circle with a little thing. You talked a little bit before when we were looking at items from the washer that you saw some possible biological material that seemed oily. Is
Yes.
this the same type of material that you were talking about?
It is.
And did you locate this material on this item of evidence, this blanket 2675
I did.
in your testing? Let's talk about that. Can you read your description of what you have, how you have this listed in your report?
Yes, so item 2675 is a blanket from the floor in the northwest bedroom. I took 2 samples from that, a scraping of red-brown staining from corner of blanket.
I don't know if I have all of it, but so you took a few samples off this one.
Yes.
Okay.
And then 2675-2, which was a sample of the possible biological tissue from the blanket.
Can you tell us what results you obtained from 2675-1 and 2675-2?
Yes. Blood was indicated on item 2675, blanket itself. Human male DNA was identified on item 2675-1. However, it was not sufficient quantity for further testing.
And that was the scraping of the red-brown staining from the corner?
Yes. Item 2675-1 and 2675-2, the DNA profile obtained from both of those items matched Victoria Martens.
And 2675-2 is one of these little pieces of material you took?
That's
an
correct.
actual sample from that?
Yes.
We heard a little bit of testimony yesterday about some swabs taken during Victoria's autopsy. So let's talk about those. Do you remember receiving about 25 or so swabs?
Yes, there were
Right.
quite a few swabs.
Is that more than normal from an autopsy?
Yes, usually. There's usually a handful, maybe 10 or so. But again, it depends on the case and what is collected by the medical investigator.
Just off the top of your head, before we get into the individual results, some of those had male DNA detected, right?
Some of them, yes.
And of those ones where male DNA was detected, how many of those had profiles that you could interpret or compare?
I don't recall the exact number. I would need to go through the items.
Okay, let's go through the items. But before we go through the items, tell me the difference between a wet and a dry swab.
A wet swab is a sample that is collected, usually moistening the swab with water. Sometimes I have seen them moistened with maybe saline or some sort of buffer, but the sample is moistened so as to collect any staining from a surface. A wet swab tends to collect DNA a little bit better than a dry swab, but a dry swab may follow or be collected routinely depending on the agency, and that is just a sterile dry swab collected of the area.
And again, that's that long Q-tip?
Correct.
On your end, is there a difference in how either of those swabs are analyzed?
No.
Let's go to your May 24th amended, May 24th, 2017 report, which is the amended of your March 28th, 2017 report.
The amended of the 28th, you said, March 28th?
Yes, March 28th
Yes.
replaced by May 24th.
Mm-hmm.
To my notes. Okay, let's talk about 4175-13. Can you tell us the description of that item?
4175-13 were neck swabs, and they were wet.
Tell us the result you obtained from the wet neck swabs.
Blood was indicated on item 4175-13. No semen was detected on item 4175-13. Human male DNA was identified on item 4175-13. Moving forward with that DNA testing.
Was that male human DNA insufficient for?
I'm sorry, yes. Human male DNA was identified on item 4175-13. However, it was not in sufficient quantity for further testing. And no DNA foreign to Victoria Martens was obtained from item 4175-13.
Is this an example of what you talked about earlier where you expect to find your own DNA on your own body?
Yes, a swab collected from an individual's body is what we refer to as a intimate swab. It was collected from the surface of the body. I would expect to find her DNA there, and I did not find any DNA that did not belong to her.
And we also had blood indicated on that swab, correct?
Yes.
Let's move to her abdominal swabs, which I believe are on your same report. So that's the May 24th, 2017.
Yes.
There should be 4175-6A, 6B, and 6C. Is that what you have?
Yes.
So just to consolidate those, tell me if you would agree that on those three swabs, you found low-level insufficient male DNA that was not interpretable, and no DNA foreign to Victoria. So kind of the same result we saw before. You found Victoria, there's blood indicated, you find DNA that matches Victoria, and then also this low-level insufficient male DNA on 4175-6A, 4175-6B, and 4175-6C.
On 6A, there was no human male DNA detected. On B and C, you are correct. It was a very low level of human male DNA that was identified, not in sufficient quantity for further DNA testing. And on all three of those items, there was no DNA foreign to Victoria.
Let's go to the swabs from Victoria's chest, which are 4175-28 and 4175-29, I believe, also contain, those are actually in the June 2nd,
Yes.
2017 report.
I'm sorry, can you repeat the number?
Sure, it's 4175-28 and 4175-29.
Yes.
Go ahead and tell us what you found on starting with 28 and then on 29.
Both of those were swabs from the chest. 28 was wet, 29 was dry. Blood was indicated on 4175-28 and 4175-29. No semen was detected on both of those items. And on item 4175-28 and 4175-29, the DNA profile obtained from both of those items matched the DNA profile from Victoria Martens.
Was human male DNA detected on both of those in insufficient levels for testing? Oh, I'm sorry, on 28, human male DNA was detected in insufficient, and 29, no human male DNA was detected?
That is correct.
Let's talk about Victoria's hand, starting with her left hand, which is on your June 2nd report, and her both hands actually are on that June 2nd report. But let's start with her left, which is 4175-15, the wet swab, and 4175-16, the dry swab.
On 41, 75, 15, and 16, blood was indicated. No semen was detected on items 4175-15 and 4175-16. Human male DNA was identified on items 4175-15 and 4175-16, however not in sufficient quantity for further DNA testing.
Go to her right hand, 4175. I have 17A and 17B, so maybe you
That is correct.
both look like wet swabs, and then we have 4175-18, which is dry.
That's correct.
So let's start with that first one, 4175-17A.
Blood was indicated on that item. No semen was detected on 4175-17A. Human male DNA was identified on item 4175-17A.
Then going to B, 17.
Okay. On item 4175-17B, blood was indicated. No semen was detected. Human male DNA was identified on item 4175-17B. And then 4175-18.
Backing up just a quick second to B, did you move forward on Y's in B, 17B?
Yes.
Tell us about that result.
Partial male Y STR DNA profile was obtained from item 4175-17B. However, no interpretation or comparisons can be made due to an insufficient quantity of DNA in the sample.
So in this one, on her right hand, you actually had human male detected and you went forward with the testing.
That's correct.
But then it was very low level. So you made the graph, you got the graph back, little boxes, and then it was just too low level to interpret.
That's correct.
Let's move to her right hand.
To 4175-18.
Dash 18, correct.
Blood was indicated on 4175-18. No semen was detected on 4175-18. Human male DNA was identified on item 4175-18. However, not in sufficient quantity for further testing.
So this one is different because on the right hand that we just talked about in your quantification stage, it looked like there might be enough to go forward. You go forward and it's still too low level. But on this one, you didn't even move forward to that Y test.
That's correct.
Your quantification says it's not going to get you anything.
That's correct.
Did you receive the fingernail clippings from the autopsy?
Yes.
And I believe those are 4175-7 and 4175-8 from the amended May 14, 2017 report.
Yes.
Let's talk about left-hand fingernail clippings, which is 4175-7.
Yes. On 4175-7, blood was indicated on item 4175-7. Human male DNA was identified on item 4175-7, however not in sufficient quantity for further DNA testing.
Let's go to 4175-8.
On item 4175-8, blood was indicated on 4175-8. Human male DNA was identified on item 4175-8, however not in sufficient quantity for further DNA testing.
And on both her fingernail clippings, you had her own DNA.
That's correct.
Is 4178 from the right hand?
Yes, 4175-8 are the right hand fingernail clippings. Ms. Archuleta, have you seen photos from the Office of the Medical Examiner of how Victoria's arms were received by them?
Yes.
And did those different arms appear to have an amount of blood on them?
Yes.
Would you expect the amount of blood that you saw in that photo to have diluted or overwhelmed other DNA that could have potentially been on her arms and hands?
There was a large amount of red-brown staining, and in my opinion, I would not be surprised to find that her DNA profile overwhelmed any DNA that might have been on the surface of her skin.
So the hand and fingernail results that we just went through where you're finding low-level amounts of nail, is that a surprising finding to you?
Not necessarily, no. I did have the information that there were other individuals in the home that were male, and so it wouldn't surprise me to find that.
But to find them in low level, if that makes sense.
Yes.
Let's talk about the left thigh, which is 4175-10, also on the amended May 24th report.
They were the left thigh swabs. It was a wet swab. 4175-10, blood was indicated. No semen was detected. Human male DNA was identified on item 4175-10, however, not in sufficient quantity for further DNA testing. No DNA foreign to Victoria Martens was found on item 4175-10.
Okay, let's go to her. The samples taken from her upper back, 4175-26, which I believe are in her June 2nd report.
Yes.
Tell us what you found on that upper back wet swab, 4175-26.
Blood was indicated on item 4175-26. No semen was detected on item 4175-26. Human male DNA was identified on item 4175-26.
Can you move forward with Y testing on this sample?
Yes, I did.
What were your results there?
A partial male YSTR DNA mixture was obtained from item 4175-26. However, no interpretations can be made due to an insufficient quantity of DNA in the sample.
So there was a mixture of, can you say how many males?
No, I have a partial male YSTR DNA mixture, but it was partial, so it was difficult to determine.
Let's go to her upper back, the dry swab, 4175-27, which is also on that June 2nd
Yes.
report.
Blood was indicated on item 4175-27. No semen was detected on item 4175-27. Human male DNA was identified on item 4175-27.
And did you go forward on, with Y testing on this?
I did. A partial male YSCR DNA mixture was obtained from item 4175-27. However, no interpretation or comparisons can be made due to an insufficient quantity of DNA in the sample.
So far, all the body standards from the medical examiner we've talked about, when you have found Y DNA or male DNA, nothing's been sufficient even when you've moved forward into Y testing.
Correct.
Okay, let's talk about the middle of her back, 4175-24, which is on your June 2nd, and then also, I think June 2nd is the main one.
Yes.
17 reports. So that's I have 4175-24, which is the wet swab from the mid back.
Yes. On item 4175-24, blood was indicated. No semen was detected on item 4175-24. Human male DNA was identified on item 4175-24.
And did you move forward with Y testing on this sample?
Yes.
What's about those results?
A partial male YSCR DNA profile was obtained from item 4175-24.
So that means it's comparable?
Yes.
And tell us about the comparisons you made.
I made the comparison to all male DNA standards that I obtained for this entire case, and every male DNA standard was eliminated from this partial male YSTR DNA profile from item 4175-24.
And that includes the defendant's standard.
That includes his standard, yes.
He was eliminated from this spot on her back.
That is correct.
Let's go to 4175-25, which is the drag swab from the mid-back. And I believe that's also on June 7th.
It is. Blood was indicated on item 4175-25. No semen was detected on item 4175-25. Human male DNA was identified on item 4175-25.
And you move forward with Y testing here too?
Yes, I found a partial mixture of male YSTR DNA of at least 2 individuals obtained from 4175-25. A major YSTR DNA profile was resolved from this mixture. All of the DNA standards, again, that had been submitted in this case were eliminated from this major DNA profile. I could not make an interpretation or a comparison regarding the minor DNA contributor to the mixture due to an insufficient quantity of DNA in the sample.
So that includes the defendant and that includes all the other standards, including her brother.
That is correct. They are eliminated.
Let's move to the lower back, the wet swab, 4175-11, which is on May 24th.
Yes. On item 4175-11, the lower back swabs that were wet, blood was indicated on item 4175-11. No semen was detected on item 4175-11. Human male DNA was identified on item 4175-11, however, not in sufficient quantity for conventional STR DNA testing, which means I did move forward with YSTR DNA testing on that item.
Conventional is the autosomal test?
That is correct. And that comparison, I believe,
6-7, maybe? Or 6-2 and 6-7?
I believe it,
And probably some others later?
yeah. I think it was before the
Oh, I'm sorry,
June
420.
report. Okay.
I have so many dates in here. 420.
The same partial male unknown YSTR DNA profile was obtained from items 4175-11 and 4175-14. All of the male YSTR DNA standards obtained for this case were eliminated from that partial profile.
So you just said, 4175-11 was the lower back wet swab.
Yes,
4175-14 is also a lower back, but this is a dry swab.
that is correct.
You said that was the same profile?
Yes,
So that's the same, so we have a partial Y,
that is correct.
and it's the same person.
Yes, it is consistent across all 4 of the samples.
Let's talk, and everybody that you had a standard for was eliminated, including the defendant.
Every male individual that I had a DNA standard for was eliminated.
I guess it doesn't make sense to test females against a Y profile. or you
Correct.
can't. Okay. That's a good clarification. So moving to 4175-14, the dry lower back swab that we just
Yes.
determined is the same person as 11, or consistent with
Mhm.
being the same person. Tell us about those results in general, which I think are on 524 and the 420.
Yes, blood was indicated on, I'm sorry, I just lost the number here.
Okay.
4175-14, correct? I'm on the wrong report. Sorry, can you tell me which report that one was on
I
again?
believe you're on the amended May 24th, and then lies
Yes,
on.
sorry, I was one ahead. Blood was indicated on item 4175-14. No semen was detected on item 4175-14. Human male DNA was identified on item 4175-14, however, not in sufficient quantity for conventional STR DNA testing, which is why I moved forward with the Y testing.
Same result on this partial profile, but comparable.
Yes.
And you eliminated all of those 38, or just the male
The male
standards.
DNA standards.
Not all 38.
That's correct.
Every male that you had was eliminated.
Yes.
Now, we've talked about 4 samples with these Y profiles. Let's look back to 4175-24, which is the mid-back wet, 4175-25, which is the mid-back dry, and then we just have the 2 lower back, 4175-11 and 4175-14.
Yes.
Are all 4 of those profiles consistent with one another?
They are consistent with one another.
So this, in your opinion, is this 1 or 2 people?
It appears to be one individual.
I'm going to go ahead and break my lunch here. All right, I'm going to keep the parties here for a few minutes, but I'm going to go ahead and release the jury. We'll be back at 1:15. All right, so the jury.
Good afternoon.
Good afternoon.
So we left off, we had just discussed the Y profile on Victoria's mid and lower back. And so at this point, that profile belongs to an unknown person.
Yes.
Let's move to let's see. So I'm sure you're aware in this case that Victoria's arms, we talked about those, and organs were found in a plastic bag in an orange laundry basket in the living room. There was a garbage bag at the very bottom of that basket, not the one containing her arms and organs, but another one at the bottom. I believe that is 1842A, and that is from your 7/19/17 report, and also some additional contained in your September 6, 2017 report.
Yes.
So that was labeled on scene as M6A, and your number is 1842A, and then you have some sub A and B.
Correct.
Okay. Let's talk about your testing on 1842A, swabbing on the outside of the trash bag. 1842-4A.
Okay,
You
sorry
wrote
about
side 1 on that.
Yes.
Just tell us, so you have a trash bag, tell us how you process this.
To my recollection, that trash bag was wet when it arrived, and the latent print analyst dried that trash bag.
I think this one is a separate one.
Is that a separate one?
So it should be 42A. But there was latent.
I'm sorry?
There was latent.
Yes.
That's what I had
So I did swab the outside of the trash bag on one side and then on the other side.
Okay, and what did you find? So side 1 is 1842-4A.
Yes.
Tell us what you found on there.
Blood was indicated on item 1842-4A.
Anything else?
Yes. Human male DNA was identified on 1842-4A, however, not in sufficient quantity for conventional DNA testing.
What about the other side? So you just said this is side 1 and then flipped over that's side 2?
Correct.
So 1842-4B is side 2. What findings did you have for that?
Blood was indicated on item 1842-4B, and human male DNA was identified on item 1842-4B, however, not in sufficient quantity for conventional DNA testing.
And then some additional testing was done on your September 2017
Yes.
report.
Yes, so on item 1842-4A, I moved forward with YSTR DNA testing of 1842-4A and 1842-4B.
And what were the results?
Partial male YSTR DNA mixtures were obtained from item 1842-4A and 1842-4B. However, no interpretation or comparisons can be made due to the presence of unresolvable mixtures.
So now let's talk about the orange laundry basket. Do you remember the large orange laundry basket?
I do.
And I believe this is going to be on your 8/18/17, which is the amendment from the May report.
And you said which one? What was the date?
8/8/17, August 8th, 2017, I think it was in the amendment, or the typo.
Yes. Okay.
And tell us what you, let's see, let's go to 1842-1. It looks like there was some staining, use of some swabs.
Yes, I did take a sample from that item of the red-brown staining and the possible biological tissue that was inside the bottom of that basket.
And tell us about the results from there.
Blood was indicated on item 1842-1.
And you move forward with PCR testing or autosomal
Yes.
stool testing? What did you find there?
Sorry, I'm just reading through.
Sorry.
The DNA profile obtained from item 1842-1 matches the DNA profile from Victoria Martens.
Did you go forward on Y testing as well?
I believe I did, yes. The partial male Y STR, it was a partial male Y STR DNA mixture that was obtained from item 1842-1. However, no interpretation or comparisons could be made due to the presence of an unresolvable mixture.
Let's talk about 1842-2, which I have labeled as a swab of handle.
Yes. On item 1842-2. No blood was detected on that item. A DNA mixture of at least 2 individuals was obtained from item 1842-2. The major DNA profile resolved from this mixture matches the DNA profile from Victoria Martens. Michelle Martens cannot be eliminated as a possible contributor to the DNA mixture from this item. Jessica Kelley and Fabian Gonzales are eliminated as contributors to the DNA mixture.
In your June 2nd of 2017 report, did you have one additional finding there as far as that mixture?
Yes, Matthew Oding cannot be eliminated as a possible contributor to this DNA mixture from 1842-2.
And then you went forward on Ys for this handle swab on your 8/18/17 report?
You said August 8th?
Yes.
Yes, I move forward with YSTR DNA testing on this item. A male YSTR DNA mixture was obtained from item 1842-2. However, no interpretation or comparisons can be made due to the presence of an unresolvable mixture.
Sticking with the items found sort of in and around that laundry basket, I'm going to show you states 93. Does that look like the orange basket?
Yes, it does.
And then we're seeing this bag in here, and I think this is the bag you were referring to a second ago that was received with some issues. So let's talk about that. So Victoria's arms and other body parts were located in that bag. What was the issue with the bags when you got them, or when your lab received them?
I got a phone call from the latent print analyst to just go observe the evidence. The bags, when he pulled them out of the packaging for his examination, were wet and had not been dried thoroughly between the layers. So he did want to just notify me, and he let me know that he was drying the items so that they would be dry when they got to me.
So tell us a little bit about that. Why do you dry wet items? Would you want to keep them wet so you could get the good stuff, or tell me how that works?
Right. So from the time that an item is collected, if the item is not dried, we don't know how long it's going to be before somebody is able to examine that item. And so if that item is sitting in any sort of conditions that are less than ideal, which is really basically anything except room temperature and dried, it runs the risk of bacterial contamination, fungal contamination, degradation of the DNA, possible other activity, possibly insect activity. And so the ideal situation in order to collect a DNA profile that is going to be able to have results that we can interpret would be to have that item that is wet initially dried thoroughly before submitting to the lab.
So because this item remains wet, is it fair to say that you would expect some deterioration of what DNA
Absolutely,
would
yes.
have been on it? And despite that, did you do a number of rounds of testing on this to see?
I did.
Let's talk about those. So you had 2 different rounds of testing on these. So let's look at 4176A, which is on the April 20th report, and 4176B, also April 20th. So this is the first attempt to get evidence from these items.
Yes. 4176A was the white trash bag, the small with the white trash bag, and 4176B was the white trash bag and disposable wipes.
Okay, so let's talk about 4176A, the results you got from that.
Blood was indicated on item 4176A. Human DNA was identified on item 4176A. However, it was not in sufficient quantity for further DNA testing.
And what about 4176B? You said this bag had some disposable wipes in
Yes.
it with it?
That's correct. And on item 4176B, blood was indicated on that item. Human DNA was identified on item 4176B, however, not in sufficient quantity for further DNA testing.
Let's talk about the second try, which is in your June 2nd, 2017 report. You tried again with 4176A.
Yes.
And did you focus on a particular portion of this? I'm going to show 93 again. I know this isn't your photo, but there were some knots tied in the bag.
Yes. When I laid that item out flat on a clean surface, I could see kind of folds or striations in the plastic where it might have been bunched up. And so I focused the swabbing of that item on the top half of the bag where the bag might have been knotted.
Is that because that's likely where someone's hands would have been touching
Yes.
the bag? And what did you find?
On item 4176A, side 1, sorry, there are a lot of numbers to go through on this one. The DNA profile obtained from item 4176A, side 1, matches the DNA profile from Victoria Martens. Everyone else was eliminated from that DNA profile.
And what about the other side? So again, you just flipped the bag and you were focusing where the plastic looked to have been
That's correct.
tied?
Yes.
What about the other side?
4176A, side 2. Human male DNA was identified on item 4176A, side 2. A DNA mixture of at least 2 individuals was obtained from item 4176A, side 2. The major DNA profile resolved from this mixture matches the DNA profile from Victoria Martens.
Did you have male DNA in insufficient quantities on both side 1 and side 2?
Let me go back and look at that. Human male DNA was identified on item 4176A, side 1, however, not in sufficient quantity for further testing. On 4176A, side 2, human male DNA was identified.
And did you push forward with Y testing on side 2?
Yes.
And what result did you get?
A partial male YSTR DNA profile was obtained from item 4176A, side 2. However, no interpretation or comparisons can be made due to an insufficient quantity of DNA in the sample.
What about the location 4176B, which is the bag that has the wet wipes in there? Tell us about the second round of testing for that second bag. And before we get to your report, tell me the condition of this bag. Was it?
By the time it got to me,
Yeah, the interior bag, was it very? I mean,
it was dried. It was very heavily coated with red-brown staining that was flaking off. And so just because it was a plastic bag and there was dried like powdery type staining, it was, I think static was kind of working with it a little bit because it was, the staining kind of went everywhere as I was swabbing.
And did that appear to be, you had a major profile for Victoria. Did you expect that was probably her blood on there?
I did, yes.
So let's talk about this interior bag with the white, side 1, which is 4176B, side 1.
Human DNA was identified on item 4176B, side 1. Human male DNA was identified on item 4176B, side 1, however, not in sufficient quantity for further testing.
And side 2?
And on side 2, human DNA was identified on item 4176B, side 2.
Was that sufficient to go forward with any type of testing?
Sorry, I'm just going
Oh,
through
sorry.
these so that I can.
I didn't know if you were done.
No human male DNA was detected on item 4176B, side 2.
Was that the entirety of that result?
Yes, there was no human male DNA detected there. Going forward with standard STR DNA testing, I'm sorry, yes, that's the end of that testing.
Okay. There was also a black sock found with all of this other evidence we're discussing, and I believe that's 1842-3A.
Is that on the same report?
Let me tell you. Should be on your July 19th, 2017 report.
Yes.
So we had 1842-3A, which I have listed as a cutting of possible biological tissue.
That's correct.
Tell us about that.
When I examined the sock, there was a piece of possible biological tissue on what would be the top of the foot area of the sock. I took a sample of that possible biological material for further testing.
And what result did you get from that cutting?
Blood was indicated on item 1842-3A. No human male DNA was detected on item 1842-3A. The DNA profile obtained from item 1842-3A matches the DNA profile from Victoria Martens.
Moving to 1842-3B, what was that?
That was a swabbing of the inner sole of the same sock
And again, why do you swab the inside of socks?
to see who might have been wearing that item.
What results did you get from that
On item 1842-3B, no blood was detected on item 1842-3B. A DNA mixture of at least 2 individuals was obtained from item 1842-3B. The major DNA profile resolved from this mixture matches the DNA profile from Fabian Gonzales.
And did you run statistics on that?
I did.
What were those statistics?
1 in 760 sextillion individuals.
Did the tissue that you just described on the sock, was that consistent with the other tissue that we saw on the brown blanket and that we talked about?
It was.
Let's talk about the items that Victoria's body was wrapped in. 4177B, which I believe is on your April 20th, 2017 report initially released.
Yes.
Okay, so what was your description of that item?
Item 4177A was a white trash bag. Item 4177B were fitted bedsheet pieces.
So let's start with the trash bag, 4177A. Tell us about the results on that, or tell us what did you test on that?
I would need to look at my notes to remember that.
Go ahead.
Okay, I'm going to have to pull them
Is
from
it
back.
hard?
Yes, please. What's that?
Is that some of the 1,800 pages?
It is, yes. It's binder number 1. I'm sorry, can you tell me the item number one more time?
Absolutely. It is 4177. I'm sorry, it might be A. 4177A.
Yes, okay. There was staining on the bag that I did test for the possible presence of blood. That was positive, and I removed a small area of that staining for further testing.
So it's presumptively positive for blood. We're talking about the garbage bag.
Correct.
What other results did you get from that?
Human male DNA was identified on item 4177A, however, not in sufficient quantity for further DNA testing.
What about 4177? And I'm sorry. Did you do PCR testing and have a result there as to that staining?
Yes, I did.
And what was the result?
The DNA profile obtained from item 4177A matches the DNA profile from Victoria Martens.
Let's move to 4177B, which is a sheet. Tell us about the results from that item.
The fitted bed sheet pieces were item 4177B. Blood was indicated on that item. No semen was detected on item 4177B. Human male DNA was identified on item 4177B, however, not in sufficient quantity for further DNA testing. The DNA profile obtained from item 4177B matches the DNA profile from Victoria Martens.
Now you tested some clothing items taken from Jessica Kelley, the defendant, and Michelle Martens. Let's talk about some of those. So starting with some items taken from Jessica Kelley when she was in the hospital, which I think are going to be on your April 20th and corrected July 20th. Oh no, so I think April 20th. So let's talk about her tank top, which is 1855B.
I'm not seeing that on this report.
So it looks like the first report was the March 3rd, 2017.
Yes, it's the May 24th, 2017 report.
So let's talk about 1855D, and you had some sub-items.
Yes.
D1, how did you have that listed?
Red brown staining from upper left side of shirt on the front.
And D2?
Red brown staining from the hem area of the shirt.
Okay,
Front.
so starting with the red brown stain from the upper left side of the shirt, what result did you get there?
Blood was indicated on item 1855B-1. The DNA profile obtained from item 1855D-1 matches the DNA profile from Michelle Martens.
Did you move forward on Ys for that item? And that may actually be on your April 20th.
Thank you. No, I'm not seeing it here.
For 1855D-1?
Oh, I'm sorry, yes, it's there. Yes.
What result did you get when you moved forward on Y testing on that?
A partial male YSTR DNA profile was obtained from item 1855D-1. However, no interpretation or comparisons can be made due to an insufficient quantity of DNA in the sample.
Let's go to 1855D2, which you said was a staining from the hem area of the shirt. So moving back to that, it was May.
What was the date of the first report?
The first part was March 3rd, and I think we were posted for the May 24th amendment. I have my amendments aligned correctly.
Yes. Blood was indicated on item 1855D-2. Human male DNA was identified on item 1855D-2. The DNA profile obtained from item 1855D-2 matches the DNA profile from Michelle Martens. And when I moved forward with male YSTR DNA testing, a partial male YSTR DNA profile was obtained from item 1855D-2. However, no interpretation or comparisons can be made due to an insufficient quantity of DNA in the sample.
Let's move to the black jeans 1855F that belongs to Jessica Kelley. Did you find it?
Yes.
Okay. And that is 1855F, and you have an F1 and an F2. So just let's start with F1. Tell us what that is described as and what you did.
On item 1855F-1, the item was red-brown staining from the left leg of the jeans. Blood was indicated on item 1855F-1. Human male DNA was identified on item 1855F-1, however not in sufficient quantity for conventional STR DNA testing. The DNA profile obtained from item 1855F-1 matches the DNA profile from Michelle Martens. Sorry, I'm trying to find the YSDR DNA test.
Should be on 420.
Thank you. That was too far. And it is 1855.
1855F-1.
A partial male Y STR DNA profile was obtained from item 1855F-1. Fabian Gonzales and any paternal male relative cannot be eliminated as a possible source of the partial male Y STR DNA profile obtained from this item.
And 1855F-1, that was on the left leg of that pair of jeans?
Yes.
Let's move to 1855F-2, which is the right leg. And was there another staining?
Yes, it was red-brown staining from the right leg of the jeans. Blood was indicated on item 1855F-2. Human male DNA was identified on item 1855F-2, however, not in sufficient quantity for further DNA testing.
What other results did you obtain?
A DNA mixture of at least 2 individuals was obtained from item 1855F-2. The major DNA profile resolved from this item matches the DNA profile from Michelle Martens.
And was there anyone that you could not eliminate from that mixture?
Yes, Victoria Martens cannot be eliminated as a possible contributor to this DNA mixture.
If she cannot be eliminated, what does that mean?
In my opinion, she is included as a part of that DNA mixture.
There were also some swabs of Jessica Kelley's hands taken at the hospital. And I believe on your August 8th amended, we have 1852S-1.
Yes.
So that's her right hand?
Yes.
Tell us what results you got from that.
On item 1852S-1, blood was indicated. A DNA mixture of at least 2 individuals was obtained from item 1852S-1. The major DNA profile resolved from this mixture matches the DNA profile from Jessica Kelley.
Did you eliminate anyone from that major profile?
Yes, Fabian, Michelle, and Victoria were all eliminated, as well as the other DNA standards that I had.
Everybody else?
Yes, and no interpretation could be made regarding the minor DNA contributor to that mixture.
And it was an insufficient profile?
Yes.
What about, was there a male YSTR profile? Did you report on Y?
Yes, there was a partial male YSTR DNA profile obtained from item 1852S-1. However, no interpretation or comparisons can be made due to an insufficient quantity of DNA in the sample.
Let's go on to let's talk about Jessica Kelley's left hand, which is 20, oops, I'm sorry. So there were some additional swabs of Miss Kelley's hand. So there is, we just talked about 1852S-1.
Yes.
Let's talk about 2682A, which is another swab of her right hand. And I believe that should be on the March 3rd, 2017 report.
The swabs from Jessica Kelley, are we referring to 2682B, the swabs from the left hand?
There's a 2682A is a different right hand swab. So let's, if we can start with that.
Okay.
What results did you get from that swab?
Blood was indicated on item 2682A. However, no human blood was detected on this item.
And what about 2682B, her left hand swab?
On 2682B, blood was indicated on item 2682B.
What about, did you find any sufficient quantity of DNA to test on 2682B?
Human DNA was identified on item 2682B, however, not in sufficient quantity for further DNA testing.
Let's talk about the hand swabs of the other people. Well, let's talk about Michelle Martens. She has a hand swab, I believe on your March 3rd report, and potentially the April 20th report. 2922A is the right hand swab.
On item 2922A, blood was indicated. Human male DNA was identified on item 2922A, however, not in sufficient quantity for conventional STR DNA testing.
Let's go forward on guys.
Yes, I did. I found a partial male Y STR DNA mixture obtained from item 2922A. However, no interpretation or comparisons can be made due to the presence of unresolvable mixture.
Let's talk about Michelle Martens' left hand, 2922B.
Blood was indicated on item 2922B. Human male DNA was identified on item 2922B, however, not in sufficient quantity for conventional STR DNA testing. When I move forward with Y STR DNA testing, a partial male Y STR DNA mixture was obtained from item 2922B. However, no interpretation or comparisons can be made due to the presence of an unresolvable mixture.
Let's talk about the defendant's hands now. That is on the July. Should be on your on March 3rd.
Yes.
So we have the left hand at 2925A. Left hand swaps. So tell us about those from the defendant.
On item 2925A, blood was indicated. No DNA foreign to Fabian Gonzales was obtained from item 2925A.
What about his right hand, 2925B?
Human DNA was identified on item 2925B, however, not in sufficient quantity for further DNA testing.
Was there a positive presumption or a positive initial test for blood? A presumptive test, I should say, for blood?
I'm sorry, yes. No blood was detected on item 2925B.
The defendant also has some clothing items collected. I think the initial report is June 2nd, or no, May 22nd. I have multiple reports listed. I think the initial is May 22nd. 2017.
So that would be the August 8th report.
I see. And remember that. Guilty. So this is item 2684B, the shorts collected.
Yes.
And you did 6 different
the report I'm looking at, I have two cuttings, 2684B1 and B2.
And I think there was some later testing that seems there
I
in
believe-
January of 2018.
Let me verify that. I think I do remember this, yes. Yes, that is correct. There are four cuttings on that one. So yes, 6 total.
So we may be able to summarize these. Was there a presumptive positive for blood on this item, 2684B?
Yes.
And I'm looking at 2684B-1. Tell us the description of B-1.
Cutting from a drop type red-brown stain on back level panel of back left panel of shorts.
And was this a mixture?
On item 2684B-1, it was a DNA mixture of at least 2 individuals obtained from item 2684B-1. The major DNA profile resolved from the mixture matches the DNA profile from Michelle Martens.
And the minor profile?
Jessica Kelley, Fabian Gonzales, and Victoria Martens are eliminated as sources of the major DNA profile obtained from these mixtures. From this mixture, no interpretation can be made regarding the minor DNA contributor to the mixture due to an insufficient quantity of DNA in the sample.
So we have Michelle Martens as the major and an insufficient minor.
Correct.
The next stain, 2684B2, I have as cutting from surface type red-brown stain on back panel of shorts. Did that 2684B2 give you a very, very similar result to what we just heard?
Yes, it did.
Okay, so that's again, Michelle Martens is the major and an uninterpretable minor.
Yes.
And then 2684B3, I listed as a cutting from top stain and vertical pattern from back left panel. Tell us about those results.
Yes.
I think that is on.
Blood was indicated on item 2684B-3. The DNA profile obtained from item 2684B-3 matches the DNA profile from Michelle Martens.
Any other results from B-3?
A partial male YSCR DNA profile was obtained from item 2684B-3. Fabian Gonzales and any paternal male relative, barring mutation, cannot be eliminated as a possible source of the partial male YSCR DNA profile obtained from this item. All of the other male DNA standards I had were eliminated as sources of this partial male profile.
There were some other clothing items recovered from apartment 407 in this case, and those are listed as items 1829, which should be on the August 8th amended.
I'm sorry, you said 1829?
Yes, 1829.
Yes.
Is that black sock?
Yes.
And I'm going to show you states 204, this is 205, and then states 204. Those items appear to be the items that you tested in this case?
To my recollection, yes.
So talking about this black sock, item 1829, what was the results? What did you test? Do you have some subs broken out into a 1B, B1 and B2?
1A and 1B. So on item 1829-1A, it was a swab of the entire outer surface of the black sock. On item 1829-1, which was the black sock, blood was indicated. No human DNA was detected on item 1829-1A. No human male DNA was detected on item 1829-1A.
And 1B?
Again, blood was indicated on 1829-1. I move forward with testing that for a DNA profile, and I obtained a DNA mixture of at least 2 individuals from item 1829-1B. The major DNA profile result from this mixture matches the DNA profile from Fabian Gonzales.
And the minor profile?
Jessica Kelley, Michelle Martens, and Victoria Martens were all eliminated as contributors to the DNA mixture obtained from this item.
Is that for the major profile?
That's for the mixture. They're eliminated from the mixture.
And did you move forward with wives?
Yes. A mixture of male YSTR DNA of at least 2 individuals was obtained from item 1829-1B. Fabian Gonzales and any paternal male relative cannot be eliminated as a possible source of the major male YSTR DNA profile resolved from this item. A minor unknown male DNA profile was resolved from this DNA mixture.
Let's move to some of the, back to the apartment and some of the other swabs. Let me see if I can kind of consolidate some of these swabs because there's a number of them. So, 2678, and they're labeled corresponding to the scene marker. So B1, B2, I think we saw some of that earlier. So for B1, B2, B3, B4, B5, B7, B8, B9, B10, B11, B18, B20, and B21, do those all come back to Michelle Martens?
Which report are we looking at?
I'm looking at a few, so let me.
Sorry.
That's okay. I think mostly the 1917 report. So I can read them off. So B1,
Yes.
just to kind of consolidate the results.
Yes.
And was that presumptive for blood?
Yes, it was positive for the possible presence of blood.
And the source of B1 is Michelle Martens.
Yes.
For B2,
Also Michelle Martens.
also presumptive for blood.
Yes.
B3,
Yes, also Michelle Martens and presumptive positive for blood.
B4,
Michelle Martens and presumptively positive for blood.
B5,
Same result.
B7. And that may be on your 420 report.
I think so. Presumptively positive for blood.
B8.
Same.
And I'm sorry, back up to B7, that was also Michelle Martens. Was the source?
Yes.
Okay, B8.
Same.
Michelle Martens and a presumptive positive for blood.
That is correct.
B9.
presumptively positive for blood. Also, let's see, sorry. Also Michelle Martens.
B10, I believe, is a mixture, so tell us about that.
Presumptively positive for blood. A DNA mixture of at least 2 individuals was obtained from item 2678B-10. From the major DNA profile resolved from this mixture matches the DNA profile from Michelle Martens. No interpretation or comparisons can be made regarding the minor DNA contributor to the mixture. Everyone else that I had a standard for was eliminated as a source of that major profile.
B11,
Presumptively positive for blood.
B12, actually, let's skip B12 for now. B18,
Presumptively positive for blood on 2678B-18. A DNA mixture of at least 2 individuals was obtained from item 2678B-18. The major profile resolved from the mixture matches the DNA profile from Michelle Martens. No interpretation could be made regarding the minor contributor to the mixture due to an insufficient quantity of DNA in the sample. And all of the DNA standards I had were eliminated from the major.
B20,
Presumptively positive for blood. The DNA profile obtained from item 2678B-20 matches the DNA profile from Michelle Martens. Everyone else has been eliminated from that DNA profile.
and that was which one? B,
2678B-20.
okay, so then B21, the last one, in this little group.
Presumptively positive for blood. And also, this profile matched the profile from Michelle Martens.
So let's talk a little bit about some of the other swabs in the apartments, whether these B dash whatever they are. We heard some testimony from Detective Pisano that little crystal violet, or something called LCV, was used to enhance some of the blood in the apartment. Those items B23 through B29. So let's take a look at B23 through B29 and summarize what you got. I believe B23 is on 420, as well as B27 and B28 and B29. And the 24, 25, and 26 are on July 19th. So we can start with 420, and we'll come back to these middle ones here.
Okay.
So B23, just while you get there, well, I'll let you look first, and then talk about it.
We'll take a break after this, ladies and gentlemen, and stretch our legs a little bit, okay? After this grouping.
Okay.
So B, or 2678B-23, what was the result there?
Sorry, I have to find where I went again.
Okay.
Blood was indicated on item 2678B-19, is
23.
23? On B-23, yes.
Okay. And what result did you get from B-23?
No human male DNA was detected on item 2678B-23, and no human DNA was detected on item 2678B-23.
Okay, let's go to B-27, which should be on the same report,
On which report?
on the same one, April 20th.
Yes.
So B-27,
No blood was detected on item 2678B-27.
B-28, same report.
No blood was detected on 2678B-28.
About B-29.
Blood was indicated on item 2678B-29.
What else about B-29 and the results?
No human male DNA was detected on item 2678B-29. And no human DNA was detected on item 2678B-29.
Okay, so moving to the last three I talked about, which are on your July 19th report, B-24, 25, and 26.
Yes.
B-24.
No blood was detected on item 2678B-24.
B-25.
Blood was indicated on 2678B-25.
What are the results for 25?
No human DNA was detected on item 2678B-25.
And 26.
Blood was indicated on item 2678B-26. No human DNA was detected on item 2678B-26.
Thank you. Let's take a 10-minute break. All rise for the jury.
Samples B-23 to B-29, as we heard from participants on over, LCD at the scene. Are you generally familiar with LCD?
Very generally, I really don't know too much about it.
So my question to you is that on some of these samples, we had blood indicated, no human male DNA, no human DNA, or on some, no blood detected. So how did we get a positive presumptive result at the scene with LCD, and then not anything really from your actual testing?
Right. So LCD testing, to my knowledge, is a presumptive tool for looking for if blood might be present. And in my experience in the field, when something like luminol is used, for example, or Blue Star, which is sort of a similar sort of screening, any staining that they're looking for happens to be in very low quantities already. And so I know when it comes to luminol, that testing reagent is a liquid, and so it can dilute what is already in very low amounts already. Again, I don't know the details of LCD, but just based off of what I know as it being a similar sort of tool, it wouldn't surprise me if that were the case, that it might have diluted something that was just already not there in very high quantities.
What if, are these the type of results you might expect if an area had been cleaned or scrubbed?
That would be reasonable to conclude, yeah.
Going back just a little bit, let's go back to 417513, which is on your supplemental from May 24th.
Okay.
That was Victoria's body swab from OMI from her neck.
Yes.
And the result was human male DNA, and when you went forward on PCR, you had no DNA foreign to Victoria. How do you reconcile seeing human male DNA and then nothing foreign to Victoria? That doesn't seem to make sense.
Well, human male DNA was identified on item 4175-13. However, it was not in sufficient quantity for further DNA testing. So that indicates to me that there's a very, very low amount of male DNA present.
So when you went forward on actual testing, all you saw was Victoria.
Correct.
Let's move back to the inside of the apartment. We're going to look at 2678B-12, which I think is contained in your July 19, 2017 report. And then if you how 2678B12 was labeled for you, or how you how it was listed in your report.
2678B-12.
Yes.
It is listed as swabs abstaining from northeast corner of kitchen table.
A kitchen table?
Kitchen table.
And what results did you get from that?
Blood was indicated on item 2678B-12. Human male DNA was identified on item 2678B-12, however, not in sufficient quantity for conventional STR DNA testing. The DNA profile from item 2678B-12 matches the DNA profile from Michelle Martens. And a DNA mixture, a mixture of male YSTR DNA of at least 2 individuals was obtained from item 2678B-12. Fabian Gonzales and any paternal male relative, barring mutation, cannot be eliminated as a possible source of the major male YSTR DNA profile result from this item. Matthew Oding is eliminated as a source of the major male YSTR DNA mixture obtained from this item. No interpretation or comparisons can be made regarding the minor contributor to this YSTR DNA mixture due to an insufficient quantity of DNA in the sample. All other standards that I had were also eliminated.
So in that Y mixture, we have the defendant, and then we have a minor profile that indicates another male is present, but there's no way to tell who it is. Is that
Correct.
Let's look at B13, which is 2678B13, which is on your April 20th of 2017 report. And if you'll just tell us how this one is labeled also.
You said 2678B-13.
13.
Swabs abstaining from south door jamb of northwest bedroom.
What results did you get there?
Blood was indicated on item 2678B-13. The DNA profile obtained from item 2678B-13 matches the DNA profile from Michelle Martens.
Was there a bias here testing?
Yes.
June 2nd report?
A partial male YSTR DNA profile was obtained from item 2678B-13. Fabian Gonzales and any paternal male relative cannot be eliminated as possible sources of the partial male YSTR DNA profile obtained from this item.
Let's go to B17, 2678B17, and tell us how you have that listed.
Swabs abstaining from door of northwest bedroom.
And what is your result from that swab?
Blood was indicated on item 2678B-17. Human male DNA was identified on item 2678B-17. The DNA profile obtained from item 2678B-17 matches the DNA profile from Michelle Martens. And a partial male YSTR DNA profile was obtained from item 2678B-17. Fabian Gonzales and any paternal male relative cannot be eliminated as possible sources of the partial male YSTR DNA profile obtained from this item.
There were some books that were part of this case. Do you recall those?
I do.
And let's look at 1853A, which I believe is on your April 20th, 2017 report.
Yes.
And can you read the description of that item for us? What was that?
On 1853A, the book is an Anchor for the Soul book.
And results on that book.
No blood was detected on item 1853A.
And on 1853B, what is the description of that?
From Faith to Faith, a Daily Guide to Victory book.
And results?
Blood was indicated on item 1853B. Human DNA was identified on item 1853B, however, not in sufficient quantity for further DNA testing.
So let's go back into the swabs from the house. There's a few places where Victoria's blood was found. Let's look at B14, which is on your April 20th report, 2678B-14, and read the description of that item, please.
Swabs of staining from closet door in northwest bedroom.
What are your results for B14?
Blood was indicated on that item. Human male DNA was identified on item 2678B-14, however, not in sufficient quantity for further DNA testing. The DNA profile obtained from item 2678B-14 matches the DNA profile from Victoria Martens.
Let's go to B6, also on 420, so 2678B-6.
Swabs of staining from kitchen floor by sink.
What was the result there?
Blood was indicated on that item. Human male DNA was identified on item 2678B-6. The DNA profile obtained from item 2678B-6 matches the DNA profile from Victoria Martens. A partial male Y STR DNA profile was obtained from item 2678B-6. However, no interpretation or comparisons can be made due to an insufficient quantity of DNA in the sample.
And let's talk about B30, so 2678B-30.
Yes.
I'm going to show you, read your description of that one, please.
Swabs from water in kitchen sink.
I'm going to show you states 45. I'm going to zoom in a little bit. You talked a little bit about biological material on some other items. If I can make it focus. Does states 45 appear to be similar types of biological material to what we've been describing here, what was on the job blanket, also on the, not on the knife, but on the, let's see, the towel from inside the washer and the sock.
Yes.
Tell us the results from, I think you have some multiple samples here, one from the water and then from some biological material. What is the result from the water? So 2678B-30, which is from 420.
Yes. No blood was detected on item 2678B-30. No human male DNA was detected on item 2678B-30.
So, is there more?
No, that's all.
So, looking at 45 and hearing the testimony of previous witnesses that talked about this water being blood tinged, how do we reconcile from the swab that was dipped in this water, not even detecting blood?
The sample that came to me was yellowish colored. I don't know whether or not the swab had been dipped in the liquid that is reddish tinged, or if it was used to collect some of the lighter pieces that are floating in that water. Just based on my test result, it would indicate to me the latter, just based off of the color, off of the other tests that I had performed on other material that looked similar, as well as the phenonegative result.
Would, just going back generally to cleaners and things like that, if soap and water, hot water, maybe even some cleaning agents had been in this sink, could that affect the results that you're seeing from samples taken from this sink?
It could affect that, yes.
And as far as the biological material, that second sample, what was the result there?
And we're talking about?
2678B-30, I think you're going to need to go to blood items.
Yes, okay. You're talking about the retest of that item?
Yes.
Okay. 2678B-30 was retested, so it was the swabs from the water in the kitchen sink. I did not do serology on that because it had been done prior in the first round of testing. I did move forward with quantification. No human male DNA was detected on item 2678B-30. And the DNA profile obtained from item 2678B-30 matches the DNA profile from Victoria Martens.
May I approach witness room? Okay. Ms. Archuleta, I'm going to show you what's been marked by identification of states 322, I apologize. Are you familiar with that chart?
Yes.
And have you reviewed that chart?
Yes, I have.
And is that chart an accurate reflection of not all, but some of the test results we've gone over here today?
Yes.
Your Honor, I would move for admission of states 322 into evidence.
No further objection.
These 22 will be admitted.
May I publish?
You may.
So, Ms. Archuleta, we have talked about a lot of stuff today.
Yes.
What percentage do you think we've hit of all the tests that you did?
Oh, goodness.
Even half?
Maybe 25-30%, and that's a ballpark figure.
Absolutely understood.
Yeah.
So looking at this chart, states 322, you indicated that this is sort of a summary of some of the things you've gone over. Is that
Correct.
fair?
Yes.
And so on this chart, we have the blanket, the blood stain from the closet in Victoria's room, the carpeting in Victoria's room with fibers from the carpeting, the faith-to-faith book, hand wipes container, the swab of the inside, the swab of the outside, that knife that we looked at, the blood stain from the kitchen floor, B6, that was Victoria, the biological material retest that we just talked about, B30, that's also Victoria, the washing machine contents, this hand towel that we looked at, and that's on here because there were 2 samples taken, so we have A1 and then B2 here. Oh, I'm sorry, that's the blue and green bath towel. So there's 2 of the blue and green bath towel and 1 of the white towel in this photo. Then we have the Tinkerbell comforter, the green hand towel, the black sock from the laundry basket in the living room, 2 of the samples there, the possible material from the laundry basket, the garbage bag that we talked about, the blood stain on the northwest door frame, the garbage bag that was around Victoria's body, the sheet or blanket around her body, and then we have a quick little summary of some of the autopsy findings and also her lower back where we did have 1 comparable profile that was a Y. All of these things, this is an accurate chart as to what we've talked about today.
Yes.
You have a lawyer?
Other than Victoria's brother, Matthew, to your knowledge, were other male juveniles collected their DNA profiles? Do you know if the others were all adult males?
I won't know the age of the individuals typically.
Okay. Now, your test detected male DNA with insufficient material for further testing on many, many items in this case. Would you agree with that?
Yes, that's correct.
Now, many of these low-level male DNA could be Matthew himself, couldn't it?
It could be any number of different individuals, but I can't make any comparisons to them because they're insufficient.
Right. And it's not really surprising in a household where there are males, not only her brother, but Fabian was living there, perhaps biological father came by. We're not really too surprised to find other male DNA at a low level in a household with males.
I would agree with that.
Okay. And so when we see, well, low-level male, that doesn't mean her killer is touching all of these different places or objects in the house.
It means I detected a low amount of male DNA.
And that's all, right?
Yes.
Okay, fair enough. Now, 2 points, I guess there's 2 items that do point to Fabian Gonzales in this, in the summary. I thought I saw it here somewhere, but the summary that she prepared. And by the way, who did prepare that summary that's called, I've got my copy here, here's the original. It's Exhibit 322. Did you prepare this?
I did not.
Okay. And the 2 items that I picked up, and correct me if I'm wrong, is a black sock that was in the living room in an orange basket, and a blood stain on the door frame in the northwest or Matthew's bedroom.
I believe that was correct, yes.
Okay, let's go over the black sock first. Now, you tested both the inside and the outside of that black sock, right?
I tested the possible biological material on the outside of the sock and the inside of the sock, yes.
Okay. And the inside of the sock showed that there was a mixture of two or more contributors.
I believe that is correct, yes.
All right. Now, if we assume that two people wore the sock at one time or another, can you tell if Fabian was the first or the last person to wear that sock?
I have no way of knowing who was first or second to wear it.
All right. When he wore the sock, can you say, was it the same day or a week earlier? How long does DNA stay on a sock or any other object?
Well, DNA can last for a very, very long time on an object given proper storage conditions. However, I have no way of knowing when DNA was deposited on an item.
Okay. One sort of end point would be if you throw it in the wash and wash it, it's probably going to lose its DNA, won't it?
I would say that the DNA would decrease in the quantity that is there, yes.
Okay. And there was a fair amount of the quantity, enough at least for you to actually identify him to a high degree.
Yes.
And the other level of comparison is when you say, well, somebody can't be eliminated.
Correct.
And we have some of that in this case as well, don't we?
That is true.
Now, isn't that sort of like, depending on how much information you have from the profile, but it's a little bit like saying, well, the person has a last name that has the letter A in it, and there may be quite a few people with the letter A, but once you start getting all of the letters of their last name, you're almost there to where you can identify the person. Is that a fair analogy to what I can't eliminate somebody means?
It's not an analogy that I've ever used. I think I see what you're trying to say in the fact that the more letters you have, the closer you get to the name. I would say that a partial profile or a profile that has less information, if more information was found on an item of evidence, then yes, I might be able to make more of a comparison if there were more DNA on that item, but I work with the sample that was collected.
Okay, yeah, you can't make them a better sample
Correct.
than they are. Now, I'm not sure, it's my understanding the biological material was found on top of the foot, not on the bottom as if it had been stepped on something.
That's correct.
And if someone were handling biological material threw the plastic bag that had body parts into an orange basket and then threw her purse on top, and the sock was in between the purse and the bag, it's at least possible that that sock was contaminated on the top of the foot with biological material after Fabian wore the sock. Is that true?
I would say that's possible.
Okay. Now, even in your laboratory setting where you have strict protocols, we had a contamination in this case, didn't we, of some sort?
There was one, yes.
And that's, I mean, I noticed the young man that was doing his work at OMI, he wore gloves and mask and scrubs and the whole material. And the purpose of that is to try to avoid contaminating one object with another, or maybe biological material from here gets on here and you start interpreting it wrong because it was contaminated.
That is correct.
Okay, so it certainly is possible that that sock was contaminated after Fabian wore it.
Well, I don't think contamination is the proper word for that. I think what I think that you're trying to get at is that it's possible that the body tissue could have been deposited on the sock after it had been placed in the basket. Yes, I think that is reasonable. That basket had a lot of blood staining and a lot of pieces of possible biological tissue within it, and so it wouldn't surprise me that some of that could have gotten on other items within the basket.
Thank you. Now, no other item in all of the mountain of evidence that you had
It's
to,
a lot.
and I'm sure the jury appreciates that, but that's the only item that has both Victoria and Fabian's DNA on one object.
Yes.
Now, there was a black sock and white socks that were collected downstairs where Fabian and Michelle had somebody call 911. Are you familiar with that? I think your number is called 1829-1A is the black sock, and I don't remember the number of the white one. But
Yeah, I am familiar with those items, yes.
okay, so for those items, which Fabian was wearing, they had negative findings for, there certainly was nothing from, as I understand it, there was no human DNA on the black sock, but there was blood on 1B. Was that part of the black sock, or was that the inside-outside thing?
Do you know which report that's on?
I'm not sure what 1829-1A would be, but it had major, it might have been left and right sock, but the major was Fabian, and Jessica, Michelle, and Victoria were eliminated.
Yes, this is 1829-1 is the black sock, and 1829-2 and -3 are both white socks.
Okay.
Can you repeat your question?
All right, very good. My question is, as I understand it, I think it's 1B, which might be the inside of the outside of the black sock. I'm not sure, but there was a DNA mixture. The major was Fabian, and you could eliminate Jessica, Michelle, and Victoria. I think that's what you said.
1829-1B did have a major DNA profile that matched Fabian Gonzales, and Jessica Kelley, Michelle Martens, and Victoria Martens were all eliminated as contributors to that DNA mixture.
Okay.
And that was item 1B, which was the inside sole area of the sock, of the black sock.
All right, so that, like the other black sock, you had an inside and an outside,
Yes.
and you eliminated, among other people, Victoria, and Fabian is a match really to that major
Yes.
contribution.
Mhm.
Now there also happened to be a minor male, but we don't know who that is.
Let me go back to this item so I can look at it. It was a DNA mixture, so there were smaller components, but yes, everyone was eliminated from that DNA profile from all the DNA standards that I received.
Okay, so it's an unknown male on his in his sock.
There is another person in that DNA mixture, yes.
Okay, and it's a male, we're pretty sure, aren't we?
It is a DNA mixture of at least 2 individuals. The major was Fabian.
So were you running your YSTR, or was that the more traditional?
On 1829-1B?
Yeah.
I have to find that if you can
That's
give me
fine.
just a minute.
I know, I'm jumping around.
That's okay. Yes, sir, you are correct.
Okay. Now, I was a little, my notes weren't very good. You found presumptive for blood on a whole string of things, B1, B2, B3, and B, I believe, stands for blood by the Albuquerque detectives. That's why they put a B there, or?
I don't know whether or not that's the reason why. I just know that it was labeled that way.
And you've got B1, B2, B3, B4, B5, B7, B8, 9, B21, and there were probably some others. And those were all basically Michelle's blood, weren't they?
Yes, there were a large number of those items that were Michelle.
So do the labels tell us, are these blood spots that they're getting in the kitchen and a hallway and out the door, or what are all of these numbers?
These are the numbers that the law enforcement agency had on their items, and so I used their item number to move forward with my testing.
Okay, and they didn't make our summary here.
No.
So we don't have like their location or anything.
That's correct.
Do your labels give us a little hint as to where all that blood was found?
Yes, and that is listed with my items that were examined, and on that first page or multiple pages of the report is the item description as best as the agency has given to us.
Now, can you summarize that in a nutshell? Where is all of this blood of Michelle's, if you know?
Oh, I would have to go back through all of those items.
It might be worth it so that we know, is she bleeding in the kitchen or the living room or where?
Do you know which report this is on?
I don't. I know you two are going through all of those Bs.
There are quite a few on the July 19th report. And it looks like there are various locations.
Why don't you read them off to us if it's not too hard? Does some say living room?
B1, 2678 B1 says from concrete outside front door.
Okay.
And again, I'm just reading through these. Let me make sure that the ones I'm reading back are the ones that belong to Michelle.
5 are all her blood, and then you skip 6.
Correct. B2 is from floor of entryway. B3 is from floor between front entryway and kitchen. I'm sorry, that was B3. B4 is from kitchen floor by washer dryer. B5 is kitchen floor by refrigerator.
Okay, skip 6.
Yes. 6 was on a different report. The next one I have is 9, and that is from cabinets above refrigerator. 11, which is from furnace door. Excuse me. 12 is from northeast corner of kitchen table. And 21 is from south wall of living room on west side of MBR door, which is, I'm assuming, master bedroom.
Master bedroom. Okay. Very good. Now, the only other item, we talked about the black sock. The other item connected to Fabian, not to the others. Well, I guess it is connected to Michelle, is B13. And that was the blood stain on the northwest bedroom.
Sorry, I'm going to have to locate that again.
Yeah, well, I'm looking at it. It looks like there's references to Victoria's room and then references to northwest bedroom. So it sounds like that's Matthew's bedroom, but I'm not sure you would know that.
I don't know that information. I only know the quote that they,
That's
or
what
the
they
information
did,
that they listed on the
northwest
item. I'm sorry, I did find 2678 B-13. That is on the April 20th, 2017 report.
Excellent. Now, can you tell, it looks like we have Fabian's, or at least you can't rule him out, and you've got Michelle's blood all close to a light switch. Can you tell if Fabian left his DNA before or after Michelle's blood was left at that location?
No, I cannot tell that.
Now, one of the things in the summary I don't see is, wasn't Fabian's blood found on Jessica's pants?
Yes.
Okay. And that might be significant. And was that a match, or just couldn't rule him out, if you remember?
I'm sorry, it's right here on the May 24th, 2017 report. You're speaking about the
The blood of
1855F?
Fabian's that was found on Jessica's pants.
On item 1855F, correct?
Correct.
Yes. Sorry, I'm just making sure I'm correct here.
That's fine.
I'm sorry, I misspoke. Those 1855 F1 and F2 were both Michelle Martens as the major on 1855 F2, and on F1, the profile matched Michelle Martens.
Okay, so the blood on her pants belonged to Michelle, not to Fabian.
Correct.
Okay. All right. So now you found sperm cells in Michelle's panties, the ones that she was wearing. Is that correct?
29, 25, I'm sorry, what item?
You don't have a number.
Objection, Your Honor, relatives.
So, no sperm was found on Victoria's body. You would agree with that?
Yes.
And none of the swabs taken from Fabian's body after his arrest contained Victoria's DNA.
That's correct.
No other article of clothing belonging to Fabian in the master bedroom or elsewhere in the apartment had any of Victoria's DNA on it.
And that is,
Objection, Your Honor. Can I approach?
Belonged to the defendant or were labeled as his?
clothing other than what is listed on the May 24th, 2017 report?
Right, because on that one we had the socks from downstairs, the shorts, and the underwear, I believe.
I'm sorry. I'm sorry, the clothing items, I think we're on a different report. On the August 8th report, there was a set of clothing. 2684 is the item number, clothing from Fabian.
And what did you find on his clothes?
On items, let's see, item 2684A. No human, let's see, I'm sorry. No human DNA was detected on item 2684A.
Okay.
On item 2684B, I took two cuttings from that item. I labeled them as 2684B-1 and B-2.
Right, and that's the one where you actually had a total of 6 cuttings. as I
Yes,
understand.
because that item I did look at it a second time on a different report.
Right. And let's summarize that one first before we move on.
Okay.
Michelle Martens was the major contributor, and Fabian couldn't be eliminated as the minor contributor on his own clothes.
He was eliminated as the source of the major profile,
Right.
and there was no interpretation regarding the minor contributor due to an insufficient quantity.
Okay, and that was B2. On B3, was he able to be eliminated?
This one is on a different report.
Oh,
I'm sorry, I'm going to have to find that report unless you have the...
I don't know which date.
Okay, that would be on the January 29th, 2018 report. 2684B you are referring to?
Yeah.
And 2684B-3, I think is what you asked about,
Right,
correct?
B3, I thought he was not eliminated as a minor contributor on his own clothes.
The DNA profile obtained from item 2684B-3 matches the DNA profile from Michelle Martens, Jessica Kelley, Fabian Gonzales, Victoria Martens, and many other DNA profiles were all eliminated as being the source of that DNA profile.
Okay. So to go back to my original question, trying to throw it all together, there's nothing of Victoria on his clothes.
That's correct.
Okay. Now, given what was done to Victoria's body, would you agree it's important to see what, if any, DNA is on her body as opposed to clothes and walls and everything else?
I would agree.
Now, YSTR is a little bit of a biased test because it only looks for male DNA, not female DNA, right?
Well, I wouldn't say it's a biased test. The reason for doing the global filer testing to begin with, the STR testing that looks at all of the DNA, looks for both male and female. The YSTR DNA testing is a tool that is used just to see that minor male component that is overwhelmed by any female DNA that is present.
And it's a helpful tool, especially when males commit a crime on a female, because the female's DNA is going to flood your test. And this YSTR, it doesn't look at female, it just looks at male.
The YSTR DNA test only looks at the male information, and it is helpful to see that male component when there is an overwhelming amount of female DNA.
It's not helpful as a tool if it's a female committing a crime on a female, because you're eliminating not only the victim but also the perpetrator.
Well, females don't have a Y chromosome, and so we cannot do the YSTR DNA test for females.
Okay. Now, yesterday we heard of all the places on the body where dry and wet swabs were taken. Let me ask you this. Were you ever asked, and it's not a trick question, but were you ever asked to analyze a pair of pink pajamas?
Not to my recollection, they were not dropped off at the lab.
Okay. Now, on 4175-13, Victoria's neck,
Yes,
I believe you said you didn't find foreign DNA, and that means DNA other than Victoria herself.
that is correct.
As I understand it, there were minute amounts of DNA on several places, but only a couple of places were there enough male DNA for the purposes of comparison.
That's correct.
Now, what I thought we would do is, can you pull up 4175-11 and we can write down what the alleles, at each loci, what the numbers are?
You're asking me to pull up my electropherograms?
The pherogram, unless you have a summary or something,
I have the electropherograms with me.
let's do it that way. I may have a copy as well. Do you have that 4175-11?
Yes, I just need to find it.
Okay, understood. I thought I had a copy up here, but we'll go with yours.
Okay.
All right, let's write down the number, and so we can orient the jury a little. These loci on the left, what are they?
The loci on the left are the pieces of DNA that we are looking at on the male chromosome.
And this is what you focus on in every DNA test, because you're looking for a match between two. If they have all the same numbers, then it's a match.
Could you rephrase your question, please?
These different locations,
Yes.
you have an electropherogram where you get a spike,
Yes.
and you get a number like a 13
Correct.
or a 20,
Mm-hmm.
and then you compare that spike and that number with a spike and a number that's an unknown sample. We don't know who left their DNA on the second sample.
Well, my job is to look at the item of evidence in the DNA profile on the item of evidence and then compare it to any known standards that I have.
Well, let's do that here, where we're going to compare the two major DNA with Gonzales. And before we get to Fabian's DNA, how do you get his numbers? We call it a known profile, but how do you know that it's his profile?
Well, You're talking about the DNA standard that was collected from
The
him?
standard, there you go.
Yes, the law enforcement agency collects the DNA standard, and that is done with a proper chain of custody. So it is known that that sample was collected from that particular individual.
And so we would call this dash 11 and dash 14, they're kind of unknown profiles because we don't know who left those.
Let's go ahead and identify that exhibit. So
Yeah.
I think defendant's M?
M as in Michael.
Yes.
Thank
I
you,
think that's
Jeff.
what we're at.
I'll put a sticker later, but, well, anyway, let's go through the first, which is 4175-11, and you're looking at your electropherogram. I'll write down the number that goes with each location. What is the number for 576? DYS, I guess, DYS 576. Is it 15?
We don't have a 576 on here. This was done with Y-filer testing, and in between some of the tests that I performed at the lab, we moved over to a different, it's the same kit, but it is an updated kit with more information.
Okay. Should we put an NR there, or what should we do with it?
Yeah, I would just leave that blank.
Okay, so we'll just leave it blank. How about 3891,
13,
448?
that is dropped out, so there's no information there.
38911,
That's also dropped out.
how about number 19?
It is a 14, but it is below threshold information.
So should we put an X line there?
You can put it in parentheses so that we know that it's below threshold.
Okay, and what's,
Trevor, I would object. I would ask that it be struck out if it's below threshold, meaning it's not comparable.
okay,
All right, let's go ahead and start that one out. Is there an objection to that, Mr.?
no, that's
All right,
fine.
let's go ahead and start that one out.
How about 391,
11,
481,
you said 481, correct?
481,
That one is also with the newer kit, I believe.
549. And I'm going in order, I think, of your electropherogram, aren't I?
Yes, but with the switch over from the
I see.
older kit to the newer kit, they changed up a little bit. That one's also on the newer kit, sorry.
How about 533?
Also on the newer kit.
438?
That's also below threshold, so we can strike that.
437?
Same with the last.
570?
Also on the newer kit.
635?
23,
390? 24?
yes, 24.
439? I think that's a scratch.
That's a 12.
Okay. 392?
Dropped out.
643?
On the newer kit.
393?
13,
458?
16,
Now this next one should normally, if you had a full profile, have two numbers. 385.
right. And that dropped out.
Okay, neither one of the numbers.
Correct.
How about 456?
I believe that is the first one up there that is at 15. The header's cut off a little bit, though. 15,
15, and then the last one, YGATA.
13.
Okay. Now, why do a lot drop out? Is it because we have a pretty weak profile and we just couldn't get a chemical reaction for each one of these locations?
Well, there just wasn't enough DNA in that sample to get a full profile.
Okay. The other strongest sample is dash 14. Can we do the same thing with that?
Certainly.
Do you have a 576?
No.
How about 3891?
Yes, that is a 13, also with some below threshold information there.
Okay. How about 448?
That's dropped out.
38911?
29. It's 3892. Sorry.
Instead of 11?
Yes.
And that would be true for both of them.
Like I.
I
Yeah.
stands for 2, I get it.
Yes.
Okay.
That's why I was wondering. It took me a little bit to find it because I was.
How about number 19?
19 dropped out.
391?
11.
481?
Newer kit.
549?
Also newer kit.
Now, when you say newer kit,
Mhm.
does that mean you did another test and you got the number, or?
No, when I tested this in Y-Filer. So at the time, that's what this was tested in. So the newer kit, you have locations for an updated kit that this was not run in.
Okay, so we may have more data if you reran, or what are you saying on a newer kit?
Yes, it is a newer kit. If it had been run in Y-Filer Plus, it's possible there could have been more information, but I don't know that for certain.
Okay, understood. All right, County 533.
That's also on a newer kit.
438?
Dropped out.
437?
15 below threshold, so we can mark through that.
570?
Newer kit.
635?
23.
390?
Dropped out.
439?
12 that is below threshold, so that would be marked out.
392?
Dropped out.
643?
That is also newer kit.
393?
13.
458?
16.
And then at 385?
14.
456?
15.
And then the YGATA.
Dropped out.
All right, let's go to the known sample of the defendant. If both of them dropped out, we won't get that number. So the first one we'll look at is 3891.
I'm going to have to find that profile.
Understood. It's just a few pages for those.
Sorry, I'm just looking for the global file profile. Give me a second to find that male profile.
For his, we have numbers basically for all of them, but we don't need to make this comparison. If there's nothing to compare to.
There is a profile to compare to. I'm just trying to find it. Okay.
All right. So the first one I want to fill in is 3891. Is that a 14?
It is.
How about 448?
20.
Oh, we shouldn't have filled that one in. How about 3892?
30.
391. We can jump down to that one.
9.
635,
21.
393. Oh, wait, let's do, there's two more. 390, I'm sorry.
That's okay. 390 is a 24.
24. 439.
10.
Now 393.
Sorry, they're printed very dark on my paper, so it's a little hard to see
Mine
them.
too.
It's number 13.
13. 458.
15?
15.
Yes.
That one with 2, 385. Do you have two numbers?
On 385? Yes, a 13 and a 14.
And two more, 456.
Sorry, I have the answer for that one, but I think the one that we labeled as a 15, 458, that's a different, give me just a minute. 458 should be an 18.
Okay. 4, is this one an 18?
Yeah, that should be an 18 right there.
Okay.
And then 456, the one you just asked about, should be a 15. And the last one should be a 12.
Okay. Now it looks like there's a few that he does match up with. 456, he matches up with.
Mhm.
And then 393, I think that's it. The probability is you would have one or two alleles in common with.
Yeah, that's not uncommon.
Are you able to eliminate Fabian from the two samples that we, the only two that we can compare with? There might be a third, but I think they all came to the same conclusion.
There were 4 total. They were all consistent. So the same information was coming up in most locations. And Fabian Gonzales is eliminated from all of those.
Thank you. Your witness. We move into evidence.
The black sock, 1842-3B,
Yes.
came from orange laundry baskets.
That's correct.
Did you find anyone else's DNA in that sock?
I'm going to have to look back at that. Can you refresh my memory about the report date?
July 19th.
Thank you.
And the secondary question that you'll probably answer at the same time is, was that even a mixture?
Okay. And you said it was on July 19th? Yes. And we're talking about 3B, correct?
3B,
Yes.
inner sole.
The inner sole. It was a DNA mixture. The major DNA profile matched Fabian Gonzales.
And then moving to 1829-1B, which was from the DNA 22nd report, which I think might be the August 8th updated.
That's correct.
You also tested the inner sole of that sock that was found in the trash can of a neighbor's house.
Yes.
And you were also able to match that sock to the defendant. Is that right? The inner sole?
That is correct.
And there was a question about the biological material that was found on the first sock we talked about, 1842-3B, and some question about, you don't know how that piece of what we now know is Victoria's biological material got onto that sock. Is that right?
That's correct. I don't know.
So, in addition to the hypothetical that it could have come from inside the basket, it could also come from someone cutting up her body and it getting deposited during that, correct?
That's also possible.
Let's talk about the blood on the door frame. There's blood on a few door frames, I think, but let me look too. One second. So, Mr. Aarons asked about the northwest bedroom and thought maybe that was Matthew's room. Do you know if that's Matthew's room or Victoria's room?
No.
Okay. So it just says northwest bedroom.
Yes.
So we'll have to rely on other witnesses to orient us as to which bedroom was the northwest. Is that right?
That's correct.
And looking at the results that we got from the 2678B13, which is on your April 20th report, we talked about this earlier, that this was blood indicated and Michelle is a contributor, and then you ran Y's and Fabian could not be eliminated, the defendant. Does that sound correct?
Yes.
And then there's another blood stain on the exterior side of the bedroom door to the northwest bedroom, which is 2678B-17, also in the 420 report, that also has blood indicated with a PCR or full DNA test that shows Michelle is the source. Then you went forward with Y's, and again, the defendant could not be eliminated from that YSTR profile as well. Correct?
That's correct.
You were asked if the only item with Victoria and the defendant on it was the sock with the biological material.
Yes.
And you're aware that there was another lab involved in this case?
Yes, I am.
And are you aware of all their results?
I've seen the report, but I don't do some of their same testing, so a lot of it didn't make sense to me.
So when you answered yes to that, that's just based on your reports only?
Correct.
What you got. Mr. Ahrens went through some of the swabs that we tried to summarize quickly that were all Michelle. The one, find the list. Be accurate. The 1, 2, 3, 4, 5, 7, 8, 9, 10, 11, 18, 20, and 21. And we went through those earlier, and those were all essentially Michelle as either the source or the major contributor.
Yes.
We skipped over B6. Mr. Ahrens skipped over B6. Do you know where B6 was found?
2678B-6.
Yes, 420 report.
Swabs of staining from kitchen floor by sink.
And that came back to Victoria.
I believe so. Let me just verify. Yes.
Just so that we're clear, when we're talking about YSTR, there was some implication that, or I think there was a direct question, was it a biased test or sort of unfair or skewed? But let's just walk through your process again. So when you do the testing, what's the first test that you do?
The global filer testing for autosomal DNA, which is, it covers looking at DNA from any individual that might be on an item,
Men and women?
men and women.
And then only when you see, well, I will just ask you, when do you go forward on Ys? When do you make that decision?
Often when there is enough male DNA that we are unable to see that clearly in a global filer profile, we will move forward with YSTR DNA testing to then look at that male component, since it seems to be overwhelmed by any female DNA present.
And we talked a little bit about that overwhelming potential for
Yes.
in DNA. In a situation where there would be a lot of female blood, would you expect that to overwhelm the male DNA?
That wouldn't surprise me if that happened, yes.
And is that a situation you go forward with Ys if you saw a quantity there?
Yes.
Let's talk about 417513, which is the next swab from the Office of the Medical Examiner. The wet swab, I believe that's on your supplemental amended 52417 report.
Yes.
Now there was a question that said there was no DNA foreign to Victoria on cross.
Yes.
And that result was from the autosomal testing, the first test, is that right?
Let me verify that. That is correct.
But there's also, on that same next swab, you have in your result that human male DNA was identified but insufficient for testing.
Yes.
And we talked a little bit about that on direct, about how you can have nothing foreign to Victoria, but then also see a male. Can you explain that again, just so we're very clear about what that finding means?
Certainly. So at the quantification step, our test is designed to look at both human and male DNA. Oftentimes, when there is a lot of female DNA, there's a very small amount of male DNA. So we might be able to detect that DNA as being there. However, I'm not always going to see a male DNA profile when I move forward to global filer testing.
So not only on this result, but I think there's several others. I believe one is her left thigh, I believe another one was the fingernails, and I'm sure I'm missing others, where you have in your results no DNA foreign to Victoria. That doesn't mean that seeing that male DNA means that there wasn't a male, right? It just means that there wasn't enough for you to produce a result to show you the profile that you could compare. Is that
Right. It just means that what I did identify was in very, very small quantities, and I couldn't move forward with testing.
let's move to defendants M. This was you testified on direct that you eliminated Fabian from the defendant from 417511 and 417514, correct?
Yes,
So this is just like a longer way of saying that.
that's correct.
Now we talked about some of these, I believe DYS 19. We started talking about a number, and then you said it was below threshold.
Yes.
So let me ask you this. You had a number.
Yes.
So how come you can't just compare it?
Our laboratory protocol has an established threshold, the analytical threshold, in which if the allelic number is below that threshold for how high that peak is, then I cannot use that information because that information is in the area that could be confused for the baseline noise, and so that is not reliable information, and so we leave that information out so that we're not falsely including or excluding an individual.
You also talked briefly, so let me back up. So there's a danger if you were to compare to even though it's coming up on your chart, right? You have a little box, you have the number, it looks really cool.
That's correct.
To me, but there's a danger if you compared that to an unsample, you could include someone or exclude someone incorrectly. Is that
That's correct, yes.
You talked a little bit about there being a newer kit that came online in the middle of this case.
Mhm.
Does that in any way negate the results that we've heard about you testify to today?
No.
Would that newer kit have eliminated the defendant more, and he's already eliminated?
No, he's already eliminated.
Do you have a moment here?
We have no further questions, Your Honor. All right, may this witness be excused? She may. All right. You may step down. Thank you.
Thank you.
Closing message 5. We'll bring you back at 9 o'clock tomorrow morning. Same place. Have a restful evening. All rights to the jury.